Dignitaries, as we welcome them into our auditorium. We welcome wholeheartedly, sir, to this conference. I request the dignitaries to kindly take the stage.
Hello. Hello.
Uh, Dr. Lathis. A very good morning to everyone here, respected dignitaries on the dais, eminent scientists, academicians, researchers, industry leaders, delegates, and dear students. A very warm and cordial welcome to all of you. It is my privilege and honor to welcome you to the third International Conference on Pharma Technology and Translational Development, integrating chemistry, bioscience, and regulatory for next generation therapeutics. Today, as we gather here, we stand at the intersection of innovation and responsibility, where chemistry meets biosciences and research translates into therapies that improve human life. And I am Dr. Annie Gupta, and it gives me immense pleasure to serve as your host for this prestigious inaugural session. Ladies and gentlemen, as the saying goes, well begun is half done. With that spirit, let us formally begin this academic celebration of science and collaboration. For this, I now request Dr. B.C. Das, sir, Dean Health and Allied Sciences, and Dr. H.R. Chitmesar, HOI, Amity Institute of Pharmacy, to kindly welcome our chief guest, Professor Ved Rabi Narayan Acharji, Director General, CCRAS, New Delhi. We welcome you, sir. Thank you, sir. I now request Dr. A.K. Singh Sir, Senior Vice President, AFSTIA, Amity University, to kindly welcome our guest of honor, Dr. Azadar Khan Sir, Senior Vice President, Sun Pharma. अच्छा ठीक May I now request all the esteemed dignitaries to kindly come forward for the lighting of the lamp, symbolizing the removal of ignorance and the illumination of knowledge, followed by chanting of Saraswati Vandana.
या कुन्तेन्दुतुषारहारधवला या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्कर प्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा शुक्लां
A big round of applause for Dr. Asadar Khan, sir. We welcome him to our conference. You're most welcome, sir. I now re-invite Dr. B.C. Das, sir, and Dr. H.R. Chitme, sir, to kindly present mementos as a token of respect to our chief guest, Professor Ved Rabi Narayan Acharji, DGCCRAS, New Delhi. Our token of respect goes with a big round of applause as we give the shawl and mementos to Professor Ved Rabi Narayan Acharji. I now request Dr. A.K. Singh, sir, Senior Vice President, Austria Amity University, to kindly welcome our chief guest, Dr. Asadar Khan, sir, Senior Vice President, Sun Pharma. I also request you, Dr. Singh, sir, to kindly present as a token of respect, shawl and mementos to Dr. Asadar Khan, sir. Thank you, sir. I now take great pleasure in inviting Dr. H.R. Chitme, sir, HY AIP Amity University, to deliver the welcome address. Dr. H.R. Chitme, sir.
Namaskar everyone. Pujaniya Founder President Sir, the Most Honorable Chancellor Sir, Honorable Madam Vice Chancellor, Professor Dr. Balvinder Shukla, Chief Guest of the function, Professor Dr. Vidya Ravi Narayan Acharya, DG CCRS, Guest of Honor Dr. Asdar Khan, Senior Vice President, Sun Pharma, respected Additional Pro Vice Chancellor, Professor Dr. Chandradeep Tandon Sir, respected President FSTI Dr. W. Salvamurthy Sir, our mentor and Dean, Health and Allied Sciences, Professor B.C. Das Sir, respected Senior Vice President of FSTI Dr. A.K. Singh Sir, esteemed resource persons for the conference, dignitaries honored of the dais, my dear colleagues, students, and dear participants joined online and offline. On behalf of the management and administration of AUP, and on behalf of faculty, staff, and students of Amity Institute of Pharmacy, Noida, and on my personal behalf, I cordially extend warm welcome to Chief Guest of the function, Dr. Vaidya Ravi Narayan Acharya DG CCRS, and Guest of Honor, Dr. Asdar Khan Sir, the Senior Vice President, Sun Pharma. And we appreciate and thank you, sir, for sparing your valuable time and gracing this occasion. I extend hearty welcome to Dr. W. Salvamurthy sir, Dr. Chandradeep Tandon sir, Professor B.C. Das sir, and Dr. A.K. Singh sir for gracious personal presence to grace this occasion. I would like to take this opportunity to highlight some of the milestones of Amity Institute of Pharmacy, Noida campus. The institute was established in 2007 with the B Form 60 intake Currently, we offer a B Farm with a 100 intake and most sought after M Farm programs in 7 specializations: pharmaceutics, pharmacology, pharmaceutical regulatory affairs, pharmaceutical chemistry, pharmaceutical analysis, industrial pharmacy and phytopharmacy and phytomedicine, and a full-time and part-time PhD programs in pharmaceutical sciences. This year we'll be starting 4 new PG programs and Form D to become only the institute in India which is offering all PG and as well as UG programs approved by the Pharmacy Council of India to fulfill the aspirations of young India and contributing significantly to the advancement of pharmacy education and research in India. We are maintaining the highest standards of education and research in line with the PCI norms and supporting the PCI in achieving its vision and mission. We are being ranked 18th in recently concluded NRF ranking 2025. Our faculty members are all with the PhD, and we have successfully completed numerous research projects that were funded by ICMR, CCRM, Ayush, NMPB, DST, ANRF, IBRO, UPCST, and industry consultancy projects. At present, there are 15 ongoing funded projects in AIP. As usual, so we have achieved 100% placement in the last year, and we are confident that we'll continue to do so in the coming years. The research outcomes of our graduates, postgraduate, PhD scholars, and faculty members are published in very high impact factor journals, including Lancet. We have 4 student cells: research and writing cell, innovation and entrepreneurship cell, human value and community outreach, and RX club. Research and writing cell and innovation and entrepreneurship cells are promoting peer learning and peer supportive system totally managed by the students, leading high number of quality publication by undergraduates and startups cultures in the institute. The Human Value and Community Outreach Cell is actively involved in developing the sense of belongingness to the society among pharmacy students. 2 of our faculty members, Dr. Nitin Sharma and Dr. Navni Sharma, recently presented their research project prototype in recently organized Unani Day at Mumbai, which was well appreciated and acknowledged by CCRM. We publish quarterly ISSN Index newsletter Pharma Panorama to broadcast the achievements of the institute. In today's conference, we have more than 300 participants registered from various institutes in India and abroad. 2 days PTT DCI Bar conference in its third series will be engaging through 4 scientific sessions along with 5 parallel sessions. So I am sure you will all will enjoy the poster and oral presentation sessions and also Pharma exhibition which is arranged in the Palm Court. With this high note, once again, I extend my gratitude to Honorable Founder President Sir, Honorable Chancellor Sir, and Honorable Madam Vice Chancellor for their blessings, guidance, and support as always. I also extend a warm welcome to all dignitaries, deans, directors, HYs, and my dear faculty colleagues, research scholars, and students who are registered to participate in the conference. My team, along with 70 volunteers, worked hard for the success of this international conference. It is my conviction that this 2 day conference, embedded with multiple scientific sessions and poster presentation, will be both highly enriching, beneficial, and enjoyable for all participants. Before I conclude, once again, I would like to express my gratitude to everyone who joined this August gathering online and as well as offline. Thank you. Thank you very much.
Thank you, sir, for such an insightful and warm words of welcome. I now request Dr. Navneet, sir, Assistant Professor and Organizing Secretary of this conference, to kindly present a memento as a token of request, token of respect to HR Chitmay, sir. Dr. Navneet.
बाद में कर दीजिएगा सर को जाना है सर का करवा
It is now my honor to invite our distinguished chief guest, Professor Vaidya Ravi Narayan Acharya, Sir, Director General, Central Council of Research in Ayurvedic Sciences, New Delhi. Sir, may we request you to share your valuable insight? A big round of applause as we welcome Dr. Professor Vaidya Ravi Narayan Acharya.
Namaste to all the dignitaries, delegates, present in this today's inaugural session for the 2 days international conference on pharma technology and translational development, integrating chemistry, bioscience, and regulatory for next generation therapeutics 2026. My regards to the Chief Advisor, Dr. A.K. Singh, the Senior Vice President, Professor Chandradeep Tandon, Pro Vice Chancellor of University, Professor B.C. Das, the Dean of the Health and Allied Sciences, Dr. H.R. Chitme, Dr. Navneet Ji, and all the senior faculty members and their student delegates from both India and internationally. At the outset, I thank the organizer, Professor, and also particularly Dr. A.K. Singh and Dr. Sharma for their kind invitations and personal touch with me to come to this occasion, sir. Dear friends, and particularly the student community, I represent the pharmaceutics of, we can say, the Indian origin, the Ayurveda. You are pursuing, are told by Dr. Chitme, near about 7 courses, post-graduation courses are being run, and you are going to create a history from this year by pursuing different modern pharmaceutics. I am the person representing the Indian traditional system, one of the Indian traditional systems, that is Ayurveda. Dear friends, India being one of the oldest civilization country, and we have our own healthcare management since ages. If you go to today's pharmaceutics, and particularly the interventions in terms of drug, near about 40%, we all know, with all the pharmacy people know, near about 40% of the drugs today developed either from the ethnomedicinal sources or from the traditional medicines or some herbal origin. The leads we got, it may be of Indian origin or it may be of other country origin. The leads we got in pharmacy, today's pharmacy, it may be metformin or it may be reserpine or other drugs. Those are from herbal origin and herbal drug, the leads from the herbal. So now, because due to some gap or others, the drugs of Indian origin, and particularly in the pre-independence era or the post-independence era, the Indian knowledge system is not being so recognized in the line that whatever the Ayurveda pharma, herbal drugs or mineral metallic drugs are being administered, they are unscientific. And the way of administrations, everything is maximum thing that unscientific way, because we don't have that much scientific values, scientific data to support in the modern parlance, the research methodology, whatever the modern pharmaceutics do cover, that is not available with us. And we always blame that Ayurveda is not the herbal drugs origin, herbal drugs is not so much advanced, well advanced in terms of modern pharmaceutics, not following the research methodology, and we blame generally the scientists in general and Ayurveda community in particular. And we all the pharmaceutics people know that I am a scholar, I am a student of Ayurveda pharmaceutics. I persuaded my graduations, but general and master and PhD and DSc in Ayurvedic pharmaceutics. And we all know that the concept of pharmaceutics is scientific based, and the research methodology, whatever we follow, is the scientists used to do the carry out the research, not a Ayurveda physicians, or it's not a other scientists. Generally, a pharmaceutics, a scientist developed the drug, and physician used to administer that drug, and maximum development in that line. But in Ayurveda and Unani or Siddha, we always expect that Ayurveda physicians will develop all the pharmaceutics and do it. So my point of concern is here that in a management line, we have the 4C principle. Generally, a good management line is always starts with a 4C principle. The first C principle is the communications. Till now, we don't have a communications among the scientists of Ayurveda and the scientists of modern pharmaceutic science in a one-to-one basis, or in a, that's communication which the today's seminar where I am honored to be here as a chief guest. I just want to say thanks to Amity that this type of communications is quite essential to know what Ayurveda science is. In terms of that is we have a strong legacy of drugs about the drugs, and it may be in Ayurveda, the drugs are of herbal origin, metallic origin, mineral origin, or animal origin, and a single formulations, you will astonished that a single formulations contain more than 50 ingredients in many cases. In many cases, a single tablet contains 50 herbal drugs or herbo-mineral or animal origin. And the selection of that is not irrational, that is rationality. Many informations are available in that we have to, until and unless we do communicate each other, and then other scientists don't know where to do research and how to do research at that aspect. Why I am saying this? Because we know the present science says the reductionist approach, that is the single molecular approach, which Ayurveda fundamentals doesn't believe in that. Ayurveda believes in the whole drug approach, and we here the synthetic, we have to find out a single molecule and do the research, target-oriented research. In that way, we are trained. From the graduation level of modern pharmacy, we are trained in that aspect. It may be a pharmacy, or it may be pharmacognosy, pharmaceutics, or pharmacology. We are being trained in that aspect, and we do submit the projects with the collaborations or funding project in that aspect. We'll do the extractions, we'll do the molecular analysis, and we'll find the single molecule and do the target study in that aspect. But the mandate of ours is different. We do admit the whole drug approach, and that is the thing difference between a modern pharmaceutics drug style and our drug style. Now, no doubt, there are so many shortcomings that a single botanical contain near about more than 100 particles, and we have to find out a scientific way, single target, single molecule, it's quite different. But science has developed. The topic, the theme of this, how to utilize this bioscience is in that aspect, that network pharmacology. Previously, we are not in a position to find 10 years back when the concept of network pharmacology are not there. We are not in a position to know all these things also. Now science has developed, so we can develop in that aspect. The second C comes about the coordinations and cooperation. Third C is cooperations. So in that way, we have to find out, we are collaborating, then the 4C is the collaborations. So communications, coordinations, cooperation, and last stage comes the collaborations. So until and unless we know our strength and the research gap where the Ayush systems wants to do research, and where we can do research in a lot of everything, That lot of information. Just I was going through the different theme, and the second step, major is the stem cell research and other things, the so-called advanced research. You will be astonished that Ayurveda gives a reference use of menstrual blood, external application, certain disease condition. When we read this, we thought that this is unscientific. This is irrelevant, all these things when you see that they classical in the Sanskrit stanza. And Ayurveda drugs gives, Ayurveda text gives responsible of the uterine breeding or well, after post-delivery, that external application, certain, what the stem cells are now doing, what we are doing in the stem cell research as on today. There are evidences, there are instances which needs to be relooked to in the modern language, in the modern scientific parlance, how to carry out those type of information research, but everything, how to carry out research. You will astonish, Ayurveda gives a definition of only 4 stanza, 60 words. How to carry out research. All the good collection practice, good storage practice, good manufacturing practice, good clinical practice ever, all are in a single 4 stanza. Because that was the point 5,000 years back, how we don't have writing system, we don't have only to listening the things. That's why the stanzas has been compressed by the Sanskrit grammatician, and we have to decode those things. This is the time. For that, we have to communicate, we have to coordinate and cooperate in that aspect. Institutions like Amity, we are doing now. You will, as a research council, I am working in a research council which is having 30 institutes. We are working more than 40 institutions in India as a collaborative mode. We are working with near about 5 CSIR organizations, ICAR organizations, and ICMR, and other universities also. So in that way, we have to, first, we need exclusively brainstorming about the pharmaceutics in Ayurveda and in which way we can work upon. Just we are working upon, working with ICAR in one of the CFA Central Freshwater Institute at Bhubaneswar about the leach applications. These are the things we administered, the drugs in different forms. The leach applications is quite vibrant in many countries previously, but now it is not there. So in this way, we need a detailed brainstorming with the modern pharmaceutics, and we are working with near about 5 NIPOs in India, and particularly about drug development, toxicity, and particularly how the mechanistic studies. Because the scientists can work in that way to make our science understand to the modern people in the modern language. That is the need of the hour, and India is blessed to have this type of soft power. If you go to the present day of international drug things, the WHO policy of 2025-39 says about integrative medicines. The WHO integrative and translational medicines, the next health, because the next healthcare of the world is interdisciplinary, or what we call that integrative. We no doubt when we got our independence, at that point of time, we have infectious disease conditions are made our lifespan were near about 45 years. Now, the lifespan of Indian citizen is near about 70 or more than 70. So the problems are different. Well, the problems, no doubt modern synthetic drugs have developed the antibiotic, they have registered for the modern acute management there, but now the disease conditions is the chronic, the disease conditions is non-infectious disease conditions, and the lifestyle disorders are the major problems, and we are most problematic will be, we are now claiming ourselves as the most youngest country in the world. Just imagine after 30 years, we will be the most oldest, near about 40% of the population will be geriatric populations. And what are the drugs available as on today in terms of synthetic drugs to manage those geriatric populations? Now, the populations of Japan, populations where the geriatric population is high, quite high, they are trying they are coming towards this natural origin of Indian drugs of Indian origin and herbal origin in particular. It may be Chinese medicine, Chinese medicines, or it may be Ayurveda, Unani, or Siddha, all are coming to us because to manage the geriatric populations, to manage the antimicrobial resistance, AMR is another challenges as on today. So these are the areas where Minister Payush works and need help to all these things to work upon. We have different dosage forms, near about 40, 50 dosage forms we are having. We do administer medicines in the enema, that is we call as basti. We call as basti. So we need research, the scientific validations of those dosage forms. We have different other dosage forms near, as I told. In technical sessions, we can details in through a presentation mode. If time, sometimes we can discuss on this aspect, all that aspects, where modern pharmaceutics can contribute and find new lead for the world to better management of the health conditions. I thanks, and again the organizer for organizing. You, dear friends, there are two systems of coded traditional medicines across the country. One is coded, another is uncoded. India is blessed to have coded system of medicines. We have Unani, we have Ayurveda, we have Siddha, which have books published books thousands years back. The well documented. We are not that traditional system which many countries of the world are pursuing. We don't have only written informations. So these coded informations can be decoded with modern pharmaceutics, help of modern pharmaceutics scientists. When we discuss together, when we work together, that's the era, that is the future era of research, is the collaborative research, with a hope that Amity will come forward for some type, and Amity is no doubt one of the best university and doing a lot researches on that aspect, but the collaborative research where the Indian strength can definitely do better. You can imagine, dear students, if you go to the history of pharmaceutics, in the 11th century, that point of time, Indian scientists, so-called, they have utilized the most dreaded poisons as a medicines. And Ayurveda is the only science which said that every poison is a medicine. If you take the example of mercury, and one of the scientists, our scientist Nagarjuna, told that by absolute power over control over mercury, I will make the world not only out of poverty, but also by out of all the disease conditions can be managed by mercury only. And that's why he started working on the mercury and gold. We all know the periodic table, and how to convert mercury to gold and make the world out of poverty. That was invented that point of time. And also how to make the world out of disease through mercury. There are about hundreds of formulations are being used as the mercurial preparations there. All the dreaded poison, arsenic, is one of the best medicines and acute medicine management of disease in Ayurveda, and also Unani and Siddha. So all these things, we have a strong legacy, but we need research to make them applicable as on today. All the disease conditions were definitely managed that point of time with this herbal drugs. And no doubt, there are shortcomings. Every science has a strength and lacunae, and development of synthetic drugs and management of surgical things. All these things are the contribution of the today's the developed biosciences. But we need our old sciences to be revamped and to be applied in the modern parlance. To develop new dosage forms is also one of the need of Ayurvedic herbal drug, Ayurveda, Siddha, Unani herbal drug industry. So with this, again, I thank the organizer for giving an opportunity to share the dais and honor me as a chief guest of this function. And all of you, I thank for your patient hearing. Thank you. And I wish all success of the today's 2 days conference, and I hope definitely some new things come up, because being a student of traditional medicines, I used to say about the traditional medicines, but a lot of topics are being supposed to be covered under this today's workshops, and definitely something will come up, and hope Amity will throw a new light out of this today's conference. Thank you all.
Thank you so much, sir. Though you do not require any introduction, but still we have to speak about you. So Professor Vaid Rabinarayan Acharji is the Director General of the Central Council for Research in Ayurvedic Sciences, CCRAS, Ministry of Ayush, Government of India. He holds a bachelor's degree in botany and a bachelor's degree in Ayurveda, followed by MD and DSc in Ayurveda. He holds a postgraduate diploma in bioethics and alumnus of Utkal University and Gujarat Ayurveda University. He has a strong academic foundation in plant sciences, biomedical ethics. He previously served as at the Institute of Teaching and Research in Ayurveda, Jamnagar, where he held key positions, including professor and head of Dravyagun, director of IQAC, director of pharmacy, director of projects, and dean. His academic leadership has shaped postgraduate and doctoral research ecosystems in Ayurveda. He has extensive experience as principal investigator of multiple funded research projects supported by national agencies and collaborative institutions. A big round of applause for Sir, and we really thank you, sir. You gave us time and graced the conference with your presence. It is now time for a special segment of our conference, the release of the souvenir. So I request all the dignitaries on the dais to kindly release the souvenir for this conference. I request Dr. Hemlata ma'am to kindly come on the stage. A big round of applause, everyone, as we release the souvenir for the conference. This shall be followed by the release of newsletter of Amity Institute of Pharmacy, Amity University. I request Dr. Hemlata ma'am to kindly accompany the dignitaries for the release of the newsletter. She's the lady behind this newsletter. Due to paucity of time and prior engagements, Professor Ravi Narayan Acharya sir has to leave. I request the audience to kindly give a big applause for sir because he graced the occasion and spared his time, his precious time, to be with us. Thank you so much, sir. Moving further, may I now invite the chairperson of PTT DICBR, Professor Dr. B.C. Das Sir, Dean Health and Allied Sciences, to share his visionary address. Dr. B.C. Das Sir.
Very good morning, one and all. Most respected Founder President Dr. Ashok Chauhan, who is a great visionary, as you all know, and creator of this great university, Amity University. He could not attend physically, but must be attending online. And very much, thank you, sir, very much for your attention and attending this online, hopefully. Our Honorable, most dynamic Chancellor, Dr. Atul Chauhan, Honorable Madam Vice Chancellor, Professor Dr. Balwinder Shukla, and the additional Pro VC, Professor Chandradeep Tandon. He is not here because today there are two inaugurations going on in biotechnology conference. That is also international conference, and this is an international conference. Same day, two inaugurations. And this week is full of lot of activities. So, that's why they are not here, and Honorable Vice Chancellor is also not here, and she will not be, I do not think she would be online. She told she will be online. So, and Dr. Tandon, and also Dr. W. Selvamurthy, who is the president of STIP and the chancellor of Chhattisgarh, Amity University, Chhattisgarh. So he is also there, but he is also supposed to be here. So, and then we have the Dr. A.K. Singh, who is the senior vice president of AFSTIA. So he is here at this moment. He has gone to leave our chief guest. So, and unfortunately, our chief guest, Dr. Jitendra Kumar, who is the managing director of BIRAC, who is the BIRAC, which is, you know, very well known organization of the DBT. So he could not come. He is traveling. So that is why our guests of honor become the chief guest. So Dr. Acharya, so this Vaidya Ravi Narayan Acharya, so he has just addressed you, and thank you for his very visionary and very excellent address. And but now the guest of honor is our Dr. Asdar Khan, and welcome you, Dr. Khan, and he is the senior vice president of the corporate relations in the very close organization related to the pharmacy or the drug development and all this Sun Pharma Pharmaceutical Industries Limited, New Delhi. So we will be listening to him, and thank you very much for joining us for this this conference. I also welcome all the esteemed speakers who has come from different places of the country and abroad. And I understand there is a speaker from my alma mater. I do not know him, but if he is here, he is from the Heidelberg DKFZ, Deutsches Krebsforschungszentrum. Here is the alma mater one sitting here, Dr. Prakash. So he is also near the adjacent institutes, what is that? The other institutes also he is associated, but he is from the same floor, the immunology floor, so of the 7th floor of the DKFZ. So you will see sometime him. So that's give me a lot of happiness that he is from my alma mater. So I welcome you all, and particularly all the delegates, speakers, and so specifically the students, faculty, colleagues, and ladies and gentlemen. I welcome you all to this two-day international conference on third international conference on pharma technology and translation development. I think it's a long title, but you should understand what does it mean. Translation development, integrating chemistry, bioscience, and regulatory for next generation therapeutic to 2026. So this is a very lot of things in this small title, 2-line or 3-line titles have been said. This two-day conference going to discuss progress and recent trends and advances in translation research in pharmacy and traditional knowledge and phytopharmaceuticals, and also to modern subjects like science of stem cells, tissue engineering, to drug development involving medicinal and pharmaceutical and molecular chemistry, then bioscience and biotechnology, and various regulatory compliances, including use of bio and cheminformatics, bioinformatics and cheminformatics, including the artificial intelligence. So I hope the artificial intelligence has a big way, and it has a huge role in the healthcare. And that's why you have to be very careful while using this technology, because it has a lot of side effects, of fallouts and bad effects. So one has to be very careful. It's a great opportunity for amalgamation of the interaction of the one side Ayurveda and Unani and biomedicine with modern biomedicine and pharmaceutical science and pharmacy and pharmacological research. I wish this conference great interactions and innovative discussion, wherein PhD students, B.Pharm and M.Pharm students must take part very actively, must ask any sense or nonsense questions even if you do not know. Since you are the student, you have the every liberty to ask any type of questions, because you are students, you have all the liberty, and which will lead to such questions will lead to some answer and discussion. And as you know, that the basis of the innovation or the innovative research is asking questions out of the box. It is not the routine questions you ask if you want to develop. So the conference on the symposia or the research conclave, etc., are organized to inculcate scientific temper and creating scientifically innovative mind in the young generation of scientists. You are the young generations of the scientists and students, and there is a lot of research opportunity and discovery of drugs and whatnot, and methods and delivery and all. So this will bring laurels, even nobody knows, some of maybe the Nobel Prizes, and who knows where one would go. So are waiting for you, and you have to come up to the expectations of the time. So that is the advice is given, not advice, but it is a true fact. So you have to come out with yourself. Nobody is less than anybody. So you have to think out of the box, and it is now transforming the pharmacy department, because it is here and there very distributed. Now they will have the new building and new laboratory, and you will have the ample opportunity for research. There are a lot of new courses have been started. So seeing the new development in the science. But with these few words, I will just wish the grand success of this conference, but I will stop here. But before that, I must thank the organizers, Dr. Chitme and Nitin Sharma and their team, and all these faculties and students, those who have involved in managing this conference, today's conference, and the national, international conference, and wish you all the best for the grand success of this conference. Thank you very much for your attention. Thank you.
Thank you so much, sir, for setting the academic tone and highlighting the importance of interdisciplinary integration, the very soul of this conference. With this, now I invite Now, to formally introduce the conference and its objectives, I invite Dr. Navneet Sharma, Organizing Secretary, PTDICBR. Sir, please enlighten us about the conference vision and thematic journey.
आपका बहुत बहुत धन्यवाद मैम और सभागार में मौजूद सभी वरिष्ठ गणों का बहुत बहुत स्वागत है. Good morning and warm welcome to the third international conference on pharma technology and transitional development, integrating chemistry, bioscience and regulatory, hosted by Amity Institute of Pharmacy, Amity University, Noida. It is an honor to be here with you today as we explore the fascinating field of pharmaceutical technology and transitional development in chem biology research. Our theme, Pharma Technology and Transitional Development, Integrating Chemistry, Bioscience and Regulatory, emphasizes the importance of collaboration across disciplines in addressing the challenges faced by the pharmaceutical industry today. From discovering new drugs to translating research into clinical applications, our discussions over the coming days will lead to groundbreaking discoveries and innovative therapies. This conference has drawn participation from various backgrounds, including pharmacy, biotechnology, life science, and chemistry, showcasing a diverse range of expertise and perspectives from around the world. We are grateful to have esteemed professionals and scholars joining us to exchange knowledge, encourage discussion to propel the field of pharmaceutical science. Before dive into the conference, I would like to extend heartfelt thanks to our sponsors, the Anshandhan National Research Foundation, ANRF, Central Council for Research in Unani Medicine, Natco Pharma, Cisco Research Laboratories, Shakti Chemicals, Progen Bio Laboratories, Sun Pharma, BioPixels, Mankind Pharma, and Fateh Education. Their support has been instrumental in making this conference possible, demonstrating their commitment to advancing scientific research and innovation in pharmaceutical science. Once again, thank you to all the sponsors for their generous sponsorship, which has played a pivotal role in bringing together research scholars and industry experts from around the world to exchange ideas and collaborate towards shaping the future of pharmaceutical science. Now, I would like to extend a warm welcome to our distinguished speakers. Your expertise and insights are invaluable contribution to this conference, and we are honored to have you share your knowledge with us. Your presentation will undoubtedly enrich our discussion and inspire us to push the boundaries of pharmaceutical sciences further. Thank you for joining us, and let us embark on this journey of exploring and collaborating together. Welcome, speakers, to the third international conference on pharma technology, transitional development, integrating chemistry, bioscience, and regulatory. Thank you, sir.
Thank you, sir, for presenting the roadmap of the scholarly gathering. Indeed, translational development bridges the gap between laboratories and life-saving therapeutics. Moving now, may I now request Dr. A.K. Singh, Senior Vice President, Amity Foundation for Science, Technology and Innovation Alliances, AFSTIA, to share his thought. Please welcome, sir. Please welcome with a big round of applause, Dr. W. Selvamurthy, sir.
Okay, namaskar and good morning to all of you. Honorable Founder President, Dr. Ashok K. Chauhan, Chancellor, Dr. Atul Chauhan, Vice Chancellor, Professor Shukla, Dr. Asdar, Senior Vice President, HR, Sun Pharma, Dr. W. Selvamurthy, Dr. B.C. Das, Dr. Naved, Dr. Chitmay, Dr. Sharma, senior faculty, students, delegates, ladies and gentlemen. I think it's a great moment for all the Amitians to be in the inaugural session of the third international conference on pharma technology and translational development, integrating chemistry, bioscience, and regulatory for next generation therapeutics.
I think just now we heard the chief guest, Dr. Acharya ji. He really highlighted or he flagged many important points, particularly mentioning the 4C, I think, which is worth practicing. And the challenge or the constraint which he shared with us is that it is time for Indians, particularly this cohort who is here, to devote some more attention and time in experimenting and validating the Ayush drugs so that we can really take our traditional knowledge or traditional systems to the optimal level of utilization. I think what I feel that this is one of the very important indication from his talk. Now, coming to the area, if we really see, as Honorable Founder President says, outcome, if we really see the outcome in health sciences in the last 20, 30 years, what is the one outcome? I will say the one outcome is that life expectancy has enhanced. The life expectancy has enhanced. And then it's now 76 years. I may be wrong by 1 year plus minus, but 76 years. On the other side, Acharya ji said that we are the youngest country. So I think this is the pride moment. We are the youngest country and we have the better the life expectancy. This is one. And what I feel, what is the shift? If you really see the shift in health sciences is we are shifting from chemicals to biologicals. And we will see a great phenomenal change of biologicals taking over as therapeutics. The other change that I feel is we are moving from reactive to preventive. Now I think we are not happy with even with the preventive. We want to move to the predictive. And the third change which these changes will bring is from generic to the specific. When I say specific means individualistic, what we say precision medicine or personal medicine or genome guided approach. So these are the changes. And if we have to take these changes further, I think the title of this conference is very, very apt. Because without integration, we cannot achieve that. And if you really see the integration is taking place. Navneet, तुमने बायोसाइंस और रेगुलेट केमिस्ट्री लिखा, मुझे लगता है इसमें इंटीग्रेशन में कुछ और भी सब्जेक्ट होंगे. इसका दायरा यहीं पे खत्म नहीं होगा, क्योंकि इंटीग्रेशन में आई थिंक वी कांट डू अवे विदाउट कंप्यूटर साइंस इन बायोलॉजिकल साइंसेस और इन फार्मेसी. I think you all would agree with me that we can't leave computer science, mathematics, engineers. इन सब का समन्वय होगा तभी जाके ये बात बनेगी. तो ये मुझे लगता है, क्योंकि इसमें बड़ी एग्जाम्पल है. बिकॉज आई वॉज इन रेडियो फार्मेसी डिपार्टमेंट ऑफ इंस्टीट्यूट ऑफ न्यूक्लियर मेडिसिन एंड अलाइड साइंसेस 40 साल पहले. I didn't know pharmacy because I have a degree in biophysics. But as I progressed, then we could utilize nuclear medicine imaging in pharmacy. And you are there, and Nitin Sharma is in the audience who had been practicing this. So integration and really made a lot of impact, what we say the pharmacoscintigraphy. Can any clinician used to think at that time that I would be able to contribute in an impactful way in the drug development or in the pharmacy? That is kind of the integration is taking place. And if we see the integration is also very much palpable if you go to any conference. I think the biotechnology biggest conferences which have taken place at Mandapam, if you see, these conferences are dominated by pharma industry. Conferences biotechnology department is the host, driven and dominated by pharma industry. This proves your thought that without integration, the things cannot move. The bio-E3 policy, I think if you really see, the researchers would be the life sciences or biotechnologists, but user or the conduit would be the pharmacy. or the pharma industry. So these things are taking place, and the main driver for this change, I think one major contribution is from the Genome Project. Because Genome Project gave us the molecular basis or genetic basis of pathophysiology, and the SNPs, which gave us that how do we differ and how can we have the personalized medicine to find new targets, because the drugs, they respond in a different way in different individuals. Now we can understand. The another technology which is impacting is the gene editing, the CRISPR-Cas9. First, we could understand that where the fault lies. Now we can correct that. We can not only correct that, we can modify that. So these are the changes which are taking place, and they are good tools in our hand, and we can really do wonders. And last but not the least is the AI, which is an overarching technology. And I feel that the most impact or the most beneficiary would be the health sciences by AI. So this is the opportune time that we make use of it. And the scope for research is quite tremendous, particularly in biomanufacturing. Do that. Either we are developing any enzyme, any drug, any peptide, any cell, and this is not only that we would be able to make them, because if you read, the cell therapy is getting a lot of approvals by FDA now. And even in India, we have got T-cell therapy available. And Hridesh Prakash is raising his hand because he's also in the queue. He had been working in that. So these changes are taking place. Not only the cell therapy, the mRNA therapy, siRNA therapy, the nucleotide therapy. So many changes, and to achieve that, we need integration. And I think these things will also bring a lot of challenges. And one would be, I would say, the identification of the appropriate target, not only the identification, then the validation or the new targets. And the another challenge which we must overcome by our knowledge and techniques is the drug resistance to of the current drugs, and understanding the targets crosstalk, and then how to regulate that. Because as life expectancy is increasing, we have compound diseases, and their management is quite difficult. But as we see, pharmacy cohort in Amity is very strong, very vibrant, because whenever I interact, I think the canvas is quite big. They have very important projects, and I am quite sure that they can make impact more palpable, more strong, and they are there under the guidance of Chitmay and Dr. Naved and all others. And Navneet had been running for quite some time to organize this conference, and he took all the interest. Let me tell you, I was going through the program. Program is very diverse, very relevant, and you have chosen good speakers. I hope they will come and they will share their thoughts, and we all would get benefited by them. And I feel that pharmacy, I will place pharmacy as a big brother. Though I am a biologist, but I feel pharmacy is at a bit higher pedestal, which binds all the sciences and pumps into the healthcare or in the health management. I think tomorrow is yours, and you have the capability, you have the capacity, and the support और the mentorship. कहते हैं ना, जो सानिध्य है, trustees का सानिध्य बहुत है, and they had been supporting, they are supporting, they will support all new ventures, all new ideas, and all new research in this area. All the best.
Thank you so much, sir. Your insights remind us that collaboration fuels scientific excellence. I now respectfully invite Dr. W. Selvamurthy Sir, President ABSTIA, AUUP India, to deliver his address. Sir, kindly address the gathering. We have been waiting for your wise words, inspiring words. Big round of applause for Dr. Selvamurthy Sir.
It's a very good morning to every one of you. Actually, I was in another important conference like this. There is an Amity Institute of Biotechnology is hosting at the I2 Moot Court. So that was looking at how do we harness the potentials of biotechnology to bring a qualitative change in our lives. So that was the theme, sustainable biotechnology applications for improving the quality of life. So I'm so happy to be here. And I was told that the addresses prior to this has been very, very mind-boggling and rewarding, inspiring. So let me also share a few thoughts. So let me, first of all, salutation to Honorable Founder President, Dr. Ashok Chauhan. For him, pharmacy, Amity Institute of Pharmacy is very close to his heart. In fact, when the university started, one of the first institutions to be started was Amity Institute of Pharmacy. So that is why he gives a lot of importance, because ultimately for looking at the prophylactic, promotive, and curative aspects, the pharmacy plays a very important life. If you look into health, these pharmacy plays a very, very important role. That's why he attaches, and we attach a lot of importance to the Amity Institute of Pharmacy. And we have very brilliant mind, Dr. Das being the dean, looking after this Amity Institute of Pharmacy, Dr. Chitme. And also we have Dr. Tanveer Naved, who has been guiding this for a very long time. And also Dr. Navneet being the convener of this, and ably supported by all the faculties of Amity Institute of Pharmacy. I'm so happy to see all of you here. And I'm also very happy to see that Dr. Acharya, who is a very important person, the Director General of CCRAS in the Ministry of Ayush, the Central Council for Research in Ayurveda and Siddha. So he was here and he addressed about how the Ayush system can actually provide a leadership role in the healthcare delivery through the pharmaceutical inventions coming from the traditional systems of medicine. So that is why I missed that address, but certainly I will listen to his the presentation, address, and all that what he has mentioned. We will try to realize it in Amity Institute of Pharmacy and Amity Universities. Now, I'm also very keen to listen to Dr. Asdar Khan, who is from the Sun Pharma, and which is a very industry very close to Amity Institute of Pharmacy. And they are supporting our pharmacy candidates in terms of internship, placement, and also collaborative projects. So thank you so much, Dr. Khan, that you are here. See, India has emerged as a pharma of the whole world. Many of them might have told. Particularly in generics, we are almost in the top, generic drugs. But then it's time for us to look into new molecule of our own, whether synthetic or coming from plant origin or even plant molecule. So we have to now look at how do we look at our own indigenous molecule, Atmanirbharta, Atmanirbhar Bharat. When you say that, you have to have your own indigenous, the new molecules coming up. That is why the conference of this type is very, very important. So we need new molecule. See, earlier, we might have taken about 10 to 15 years or even 20 years to develop a new molecule. If it is synthetic molecule, you have to have the standardized the formulation, in vitro, in vivo, safety, efficacy, phase 1, phase 2, phase 3, phase 4, post-marketing surveillance. All this would have taken 20 years. But today, it's not required to spend that much time. We are able to compress the time at which would have taken to bring a new molecule to application. So that why? How? The first is data analytics. We have huge data collected over during the almost 50, 100 years. So you have a huge database. So data analytics, and again, using the artificial intelligence, you will be able to downstream the potential molecules, predict its susceptibility, the adverse effect, the potential side effects. All this can be predicted, and that could be addressed even at the synthetic level. So that is why that now you can reduce the time duration for a new molecule to get into application. Now from Amity Institute of Pharmacy itself, a number of products are coming in the offing. I would like to mention a little later. So that is one. The second is we have our own natural products available, which has stood the test of the time. When you look at Ayurveda, a number of herbal the ingredients have been used. So how do you bring out a new drug for various applications is also another challenging, interesting, and possible areas. Then the next thing is when you look at global pharma market today is $1.75 trillion. Imagine the huge market. The pharma industry today, the market is 1.75 trillion, not billion, trillion. And India has a share of 55 billion US dollars. So that is the number today. So we want to augment this. This is Viksit Bharat, developed India. So you need to have your own molecule, new molecules, generics, natural products. All this will come and will reach a major share in the global market. Then when you look at the 20% is the global generics out of this market today, 20% comes from generics. The world market, 20% is from India in the form of generics. 60% of world vaccine is developed in India. Vaccine development, we are 60%, we hold the share. So this is our power in pharmacy, pharmacology, pharmaceuticals. This is the power India has. And you know how much we export to other countries every year? It is 30 billion US dollars, 30 billion. That is only the export of pharmaceutical product going abroad is 30 billion. So you see the youngsters, particularly students, let's give a big clap to students, come on. That is the market. Amity Institute of Pharmacy, you people are going to look at $1.7 trillion today, which is growing almost 20% the annual growth rate. So that is the market which you should now look at. So now innovation ecosystem, how do we create that kind of innovation system, which will bring a new molecule to market, to application, bench to bedside, lab to land, mind to market? How do you bring this? The first is you should not stop looking only at demonstrating the effects of certain molecule. You should now look, that macro picture should be available when you launch a project, that how will I take it to the market? How will I scale up? And how will I produce to the quality and cost effectiveness, so that all this should be there when you think of a pharmaceutical research, not only in the form of formulations, what kind of formulations I will bring. So all this should be available. So you should also have scale-up facilities, pilot scale. Don't stop at lab scale and bring it to pilot scale, then give it to industry like Sun Pharma, Dr. Khan is sitting here, and so that it ultimately goes into the production and application. Then our regulatory aspects, now that is also speeded up. Our systems of Drug Controller General of India, now they have speeded up the processes. It is still increasing its efficiency of clearing the cases for the clinical trials. So their regulatory process is again being speeded up. Now coming to Amity Institute of Pharmacy, we are number 18 in the whole country, NIRF ranking. And our university is 22 as a university. But Amity Institute of Pharmacy occupies a very important position of number 18. Let's give a big clap to all the faculties, heads of the institution, dean, who has done this honor for us. And so this is one. They have brought out some new molecule, Dawaul Kurkum. One of their faculties, Dr. Mariam Sarawat, she has brought out through CCREM a drug which is known as Dawaul Kurkum for hepatocellular carcinoma. A drug has been developed from Amity University, which has already been cleared for clinical trials. Now, actually, we are looking for a government-funded institution like AIIMS and also a private the hospitals to carry out the clinical trials. So the CCRUM is going to support this. So we have done the safety efficacy trials, now waiting for the clinical trials. So one product which is coming from Amity Institute of Pharmacy is this. Then we also have Amity Center for Translational Research. We have Dr. Hridesh Prakash sitting here. He has also been cleared for a nutraceutical trials, which is going to take place for the tuberculosis. See, we are now looking at sphingolipids and then precursor like L-serine. He is now developing a nutraceutical for the intervention, which is cleared by Indian Council of Medical Research, their committee for taking this as a nutraceutical application for tuberculosis control and management. So another second product which is coming from this portals of Amity University. Third, Dr. B.C. Das himself, his group, Amity Institute of Molecular Medicine and Stem Cell Research, is developing a triple conjugate, a known anti-tumor agent, curcumin, folate. He has made a conjugate. His group has made a conjugate, triple conjugate. It's very difficult, challenging to combine, conjugate a synthetic molecule with a natural molecule like curcumin, which they have done. Seema Bhatnagar is the synthetic chemist who has done this conjugation under his leadership. So now that is also getting into preclinical safety and efficacy trials. So what I want to say is, I just want to give Amity University not only churn out the human resource in pharmacy, but we produce the end product, which will be, you will see them getting into market soon. Similarly, Dr. Mona Lisa Mukherjee, who is heading the click chemistry, she has come out with nanohydrogel for the sustained drug delivery. So this is for the deep wounds like diabetic wounds. How do we heal? By giving a sustainable delivery. So we are working on, we have already developed this. Now it's undergoing the clinical trials that is on the nano hydrogel for deep wound healing. So I can go on telling these are exciting things happening in Amity University under the able leadership of our founder president, Dr. Ashok Chauhan. And now final message, the carry home message, takeaway is speed up the process. You know, now you can't wait 15, 20 years to develop something. Speed up the process, come out of the comfort zone. See, we should come out of the comfort zone. If Viksit Bharat has to happen, every one of us has to make sacrifice. Every one of us should contribute. So come out of the comfort zone, contribute for the speedy development of new drug, whether it is from natural source or the Ayush system or from synthetic molecules, speed up. Use advanced technologies like artificial intelligence, data analytics. All this will help you to facilitate, catalyze, reduce the time cycle, the life cycle for a new drug development. So that is another thing. And focus on translation. Very important, because we are happy with publication, filing patents. Don't stop with that. Translate them into a deliverable. So that is another message. Focus on translation research. India should assume leadership role. We have been followers. We have been assuming leadership role in certain domains. But I want in pharmacy, I want in the biotechnology, pharmacy, a leadership role for India. It will happen because, as Dr. Ashok Chauhan says, that, you know, success by compulsion. We have to succeed means we have to succeed. Success by compulsion. That is his philosophy. I'm sure this will also happen. India will become a Viksit Bharat. India will assume the leadership role in this field as well. So my best wishes, we'll be eagerly looking forward to the outcome from the deliberations and also new trajectories, new avenues for taking the deliberations forward and giving it to the students and young faculties as well as the future leaders so that they will take it forward in a fast track mode. Thank you. Best wishes.
Thank you so much, sir. We have been extremely privileged to have with us our guest of honor, Dr. Azadar Khan, sir, who heads India Corporate Relations, India Regulatory Affairs, India Clinical Research, and CSR at Sun Pharmaceutical Industries. He is an industry veteran, carries over 46 years of rich experience in pharmaceutical industry. In the domain of HR, regulatory affairs, corporate relations, quality, and compliance. He joined Sun Pharma in 1997 as part of the merger of Tamil Nadu Dada Pharmaceutical Limited, TDPL, where he had functioned for 17 years. He grew up in Sun Pharma, handling various positions and portfolios and played critical role in organization success. I can go on with your star-studded introduction, sir, but I would like to end here and welcome you to kindly come here to come and inspire us with your inspiring words. Please welcome Dr. Azadar Khan, sir.
So good afternoon to all of you. At the outset, I express my deep regard to the founder president, Dr. Chauhan, actually, who has created a institute of repute which has become a national pride today, and which is also in one way has helped the pharmaceutical industry in getting the manpower and its requirement. I am also thankful to the Vice Chancellor, the team, the organizers for giving me this opportunity to be with you and this opportunity to talk to you. I think when it comes to pharmaceutical industry, all of you know that pharmaceutical industry in India is a very fragmented industry. Almost 10,000 manufacturing units exist in India. This is very unique to India. Nowhere in the world you find so much of pharmaceutical manufacturing unit. Sun Pharma being the #1 company in India, our market share is only 9.6%. You can imagine a company with a 9.6% market share can become the India's topmost company. So you can gauge from this number the fragmentation of the pharmaceutical industries, a very highly competitive industry. We produce almost 30 billion worth of drugs for domestic consumption and the similar amount of $30 billion for exports. So our domestic consumption and our exports are almost equal. We cater to about 200 countries. And of course, there are certain challenges in meeting the stringent regulatory requirement. But in India, I think we have largest amount of US FDA manufacturing unit as compared to any other country. So I think today there is a lot of, and I also congratulate the organizers of this event for choosing the very appropriate topic for this conference, which is the translational research. Today, I think there is a lot of emphasis on translational research. Historically, we have been all scientific institutes have been focusing only on publishing papers. I think that anything which you reward and encourage, that only gets promoted. So we have been only evaluating the efficiency of the scientists by publication of papers. I don't say that publication of paper is not necessary. It is necessary, but whether the research has converted into any commercialization of any product, I think that is the very, very important question before us. If we look, the institutes like CDRI, who have huge budget, huge capability, etc. But over the years, did you see that how many drugs from CDRI got commercialized? I think Dr. Nityanand's time, one the contraceptive drug was the only which saw the light of the day. Beyond that, I think we have not seen anything which is getting commercialized. I feel that this has been an area to be focused, and I am very happy that the government has turned its attention now to focus on commercialization of the product rather than only publishing the paper and obtaining the patent. Today, we have attained a sizable amount of leadership, and we are the leader in generic medicine. Every third pill consumed worldwide is produced in India. We very proudly call ourselves as a pharmacy of the world. And at the same time, we also in the vaccine about, as Dr. Selvam said, that we almost 60% of the world vaccine is being supplied out of India. But I think if we see our ranking in volume versus value, then we are very low. We are third worldwide in producing the volume of pharmaceuticals. But value-wise, we are 14th ranked. So it means we are sending the goods in containers and we are getting the payment in envelopes. So I think this has to, if this situation has to reverse, moving from volume to value, then I think it's a clear realization that this can never be achieved only by producing generic products. The kind of targets which Government of India has set for itself and for the pharmaceutical industry, that today we are 60 billion. By 2030, we have to become 130 billion. And by 2047, Viksit Bharat, our target is to become 400 billion. So this kind of number cannot be achieved only by supplying generic to worldwide. So therefore, now the attention has to be focused on innovation. So innovation becomes a buzzword. Several meetings which we had with the Principal Secretary, Principal Scientific Advisor to the Prime Minister, Dr. Sood, etc. I think everywhere the buzzword is innovation. How we can get into innovation and how we can start innovation. And I think to start with, drug discovery, new molecules, of course, means these are all the much bigger area. But there are some low-hanging fruits also, which are called incremental innovation. I think some incremental innovations also can give us some kind of additional remunerative competitive advantage in international market. Like I will quote a simple example of Sun Pharma itself. We have, all of you know that there is a drug called gemcitabine. Gemcitabine is a non-stable drug because it's lyophilized for stability. And it was available in lyophilized form only. In liquid form, it was unstable. We saw that because when an oncologist prescribes the prescription for the patient, and especially indoor patients, he writes the prescription by calculating the body weight, and the dosage are determined as per the body weight of the patient. So certain ml is to be administered to the patient. And that reconstitution and administration is purely left to a nurse. Now, many times we have seen that there are calculation errors, there are administration errors, and if the drug is given less than the required quantity, then it is a suboptimal dose. It doesn't work. being a cytotoxic drug, if you give higher quantity, then it has a side effect. So both ways, I think the accuracy of dose was very, very important for gemcitabine. Now, with almost 3 years of research, we could stabilize this product. Now, this product is available in international market and India as well in a ready-to-use bag. So per ml is just the nurse has to administer the how many ml is to be given. So it's ready to use kind of a bag. And this has, we have got a patent for this, etc. And I think international market, this product has got such a wonderful acceptance that it has given us a very quantum jump in terms of our. So this is not something a drug discovery. This is an incremental innovation. If we see that some of the products, some of the injectable APIs, they are non-soluble. And they are soluble in a media which is oily. Now, when these injections are intramuscularly administered to the patient feels a lot of pain. So these are all painful injections. So I think how we can make those kind of incremental innovations by which we can make the life of patient more easy and we can reduce the pain of the patient. So these are all incremental innovation on which also we should be focusing and we can get some kind of a premium and leverage in the international market by bringing incremental innovations. I think I told you that we have to move from volume to value. And and I am very optimistic about pharmaceutical industry doing well and achieving these kind of targets because there are various reasons for this. Reason is one is that someone already quoted that we are increasing aging population. I think up to a age of 40 years, nobody or most of them may not be consuming any tablet. But today, hardly you find a person who is around 50 and not taking any medicine. So I think people take 6, 7, 8 medicines because there are many diabetes, hypertension, this, that, etc. So 6, 7 pill every person takes. So I think the way aging population increases, the consumption of the drug will also increase. So one reason that there would be. The second thing is that increased penetration of medical health insurance. So I think health insurance also gives a lot of affordability to the people to get the better treatment and therefore it increases the consumption of drugs. Then government introduced schemes like Ayushman Bharat, and these are all the schemes which also give an affordability of treatment for the people. Otherwise, in India, most of the people, it was out-of-pocket expenses. It was self-borne expenses by the patients. And the second thing is that our ability to diagnose complex ailments and complex diseases with digital tool, with AI, support, etc. So I think we are now able to detect and diagnose complex diseases at early stage. So these are all, I think, very important aspects through which we feel that we will be able to achieve these targets, and it's not very difficult for us to this. So now, once we look for innovation, so how the innovation happens and what would be the drivers for innovation? So I think what we see that there would be certain strategic drivers to create innovation in India. One of this would be to create a research focused ecosystem. Today, I think companies like us struggle in doing a lot of preclinical studies. The preclinical studies, we have limitations to do India. We can't do large animal studies in India. These studies are getting done outside India. Some studies we get conducted in Germany, some studies we get conducted in Australia, etc. And believe you me that these studies are conducted outside India at least 10 times or 15 times of the cost than we can conduct this in India. So preclinical studies, I think there will have to be a environment wherein the companies who have capability can do the preclinical research with large animals in India. Then I think is also one of the very important factor that there is a strategic driver in terms of the supply chain, backward integration for the products. Today, I think, thanks to COVID, that it woke up all of us that we need to have self-reliance in API because things were going on. Government introduced a lot of good schemes like PLI scheme, etc., to get self-reliance and Atmanirbhar Bharat in terms of API production. Everything means I think significant achievement have been there, but I think what is required and what is today, there is a lot of still gap exists, and we need to catch up. That needs a lot of government support to counter the competition from China, because Chinese models are very different, and we have to have some kind of a methodology that we. The second thing is that in India, in every country, the price control is there. But I think price control, like China, how they have encouraged the innovation. And if you see last 3 years, the number of patents filed by China for formulation, for the biosimilars, for biologics, they are huge numbers. And how they are able to achieve. So one is that they identified all key talent who are involved in research outside China. They brought back those scientists to their own country, and they helped them set up the kind of lab they were working. And they said that you have to work for our country. They took it in a mission mode. And this has helped the country to significantly increase the patent filing. And now number of patents filed by China. The second thing, now many multinationals company, they are doing their drug research in China, because they said that you come invest in our country in research, use my population for the clinical trial, first develop the drug for my country. We will use it in my country. And then you can subsequently register in other countries. Now, Chinese companies or innovation done in China by Chinese company. There is a lot of support by the government because they buy most of the things for the government public procurement schemes. So that way it gives a lot of incentive for the innovation. Today, I think even when innovation comes, you know that one of the product, Seriglotazan, which was one of the IND by Zydus Cadila, which was worldwide patented. Today, after having almost 5 years of introduction of this product in India, do you know what is the sale? They are not even doing 200 crore for this product. So with this, how the companies expect to put this much of money into research, and if there is no proper reward for the innovation. So like China, they procure all these things, but in our government procurement schemes, the L1 is there. So until unless L1 is there. So I think these are all the things. If we have to encourage innovations, then whole ecosystem needs to be changed so that innovation is encouraged. And I think the other thing is that the broaden global reach. You may be reading and hearing that there are a lot of foreign trade agreements are being done. So the government is also making a lot of effort to remove the non-tariff barriers. So when non-tariff barriers is removed, like the European FTA has been done by India, which is a very, very good bargain and which is a good negotiation. So likewise, I think the recently the FTAs which have been done, I think they broaden our reach to the global market. So I think these are all the very important things which we need to look when we have to give thrust on the innovation. Now, I think one of the important, and what are all the other, what could be the other enablers for us for the innovation? The other enablers could be one of the very important is this competent talent. I think where as an educational institute, you have a much, much greater role. Because we find that since we have been involved only in generic production and development of drugs in our laboratories, our work in innovative direction was very less. Therefore, we don't have very competent scientists who can do this kind of innovative work. And that talent is very different, and especially if you look into the drug development, NCE development, etc., then the 70% of the cost is in clinical development, in the clinical research. Only 30% is the laboratory and etc. So the major chunk of the cost is in clinical research. Now, when clinical research happens, today what we are struggling to find that since we have not a competent pool of scientists who are able to do clinical development, because clinical development, how to make a protocol with minimal number of subjects so that it gives us validation with the new drugs, etc. So I think these are all the talent. As a Sun Pharma Science Foundation, you may be knowing that we have been rewarding the scientists and research scholars for Sun Pharma Science Award. But from this year, we have also introduced a fellowship for the clinical research scientists, etc. So I think there has been an advertisement. People can apply for that and can get benefit of the fellowship which we are according. So I think today, if we see that educational institutes, the kind of a crop you produce from here, what is their employability? I think today, less than 20%, or even the figure could be much lesser. The government employs less than 20% or even less. So 80% of the people are absorbed in the industry. So industry, how does the industry needs a industry ready material? I think the whole focus has to be on there so that the employability of the students increases, which is possible when there is a very good connect between the academia and the industry. Because industry will know commercial aspects of the products, and the knowledge imparted by the educational institutes will give them subject knowledge. Internationally, if we see, majority of the inventions and majority of the innovations, they are coming from educational institutes. Because educational institutes have this kind of a funding in other countries where they do research, and so once the proof of concept is created, then I think industry takes it, and majority of the products, I can name many products that which have been actually invented or innovated by the research by the educational institutes and subsequently adopted by the industry. So the employability and industry academia relationship is very important. The ranking of any educational institutes also gets better or higher when you are able to place your all the candidates. So I think today every industry needs industry ready students. Some of the products, some of the courses which have been taught, they are not in sync with the current industry requirement. Maybe today in pharmacy courses, we don't teach the data science, data integrity, data traceability, all these kind of which are very important subjects. So I think our today, if you see majority of the Indian plants which have a US FDA audit observations, they are all mainly related to the data integrity and other things. So I think these are all the aspects which we have to focus and produce a talent which is ready for the industry ready, and they can be used. And I think we are very keen to develop this kind of a relationship, because we being the largest industry in the country, we can get benefited from these kind of a students, and we can drive our mission very forcefully, and we can increase our presence in the outside country. Many schemes today for the research and innovations are introduced by the ANRF is one of the scheme, the PLI scheme, the PREP scheme, which has been recently announced by the government, that PREP scheme, we have applied about seven products under the PREP scheme. So, and now recently this Bioshakti Mission scheme has also been introduced by the government. So there is a lot of support being given from the government side, and we should take advantage of this. And what I say that what we have been doing for generic medicines, I think if we have to shift our gear to innovation, then I think our whole education system and preparation of the scientists also need to be in sync with the demand of the time today. So thank you very much. Thanks a lot. And I think once again, I thank the organizers for giving me this opportunity to share my thoughts with you. Thank you so much. Thank you.
At Amity, we believe in industry, academic symbiosis, and leaders like you keep inspiring us. So thank you so much, sir, for your inspiring words. As we come to the close of this inaugural ceremony, I invite Dr. Tanveer Naved, sir, Deputy Dean, Health and Allied Sciences, Amity University, Noida, to propose the vote of thanks. Dr. Tanveer Naved, sir.
Very good afternoon to one and all. On behalf of the organizing committee of PTT DI CBR 26, it is my proud privilege to propose the vote of thanks. First and foremost, we would like to express our deepest gratitude to our Honorable Founder President, Dr. Ashok K. Chauhan sir, for his blessings, visionary leadership, and unwavering commitment to academic excellence and research driven at Amity University. Honorable Founder President guidance continues to inspire us to create meaningful platform like this for scientific advancement and serve vision and mission to contribute in the Viksit Bharat Mission 2047. Sincere thanks to our dynamic Chancellor, Dr. Atul Chauhan sir, for all the encouragement and motivation. We greatly acknowledge the guidance and support of our respected Vice Chancellor, Dr. Balwinder Shukla Madam, who could not attend this inaugural session due to some family commitment today. Our heartfelt appreciation and gratitude to our chief guest, Professor Vaidya Ravi Narayan Acharya ji, the DG of CCRAS, and his valuable insight about the Ayush system of medicine, which is one of the key, system in the country. A special thanks to our guest of honor and the industry stalwart, Dr. Azadar Khan sir, who is the Senior Vice President of Sun Pharma, and his all the valuable insight about the latest happening in the industry, especially in the preclinical and clinical research. Thank you very much, sir, for gracing the occasion. A special thanks to our mentor and senior advisor, Dr. W. Selvamurthy sir, who always inspire us with his word of wisdom, and today his address was no exception. Thank you very much, sir. A very, very special thanks to our respected Dean, Dr. B.C. Das sir, whose guidance is always, key for AIP, whatever activity, whatever event we organize. Thank you very much, sir, for all your mentoring. My special thanks and gratitude to Dr. A.K. Singh, sir, who contribute a lot for this conference, especially inviting our chief guest and guest of honor. Thank you very much, sir. Sincere thanks to Dr. Chandradeep Tandon, sir, the additional Pro-VC of AUP, and due to some parallel conference at the AIB, he could not attend the inaugural session. I would like to thank my colleague, Dr. H.R. Chitme, sir, the HOI of AIP, for all the guidance and leadership for this conference. For success of any, conference, we required one person who can guide the entire show. And luckily for this conference, we have Dr. Navneet Sharma, who is the organizing secretary of this conference. And I know right from the day one when we announced this conference 4 months ago to till date, he worked tirelessly for the one mission to success this conference. And from inviting the very quality speaker and to this inaugural session, you can see the beautifully decorated auditorium. So all credit to you, Dr. Navneet. Very well done. My sincere thanks and gratitude to all my faculty colleagues for the part of the organizing committee, and each one of you contribute, especially last 15 days, I saw that each one of you, working with whatever committee allocated to you. So round of applause for our organizing committee. I thank Ms. Parul Singh from FSTR for liaising with various guests and speakers of the session. I would like to thank all the delegates, the speakers, session chairs, the scientific evaluator of the conference, whose participation adds intellectual richness to this August gathering. Sincere thanks to all of you. Thanks to all the sponsors of the contribution for this conference for their contribution. And thanks to admin team, the IT team, the media team, and all who support this conference. Lastly, my sincere appreciation to all the student volunteers, without whom you cannot expect any conference to be successful. And I request all of you to give a round of applause for all the student volunteers who contributed to this conference. Finally, look forward for the insightful deliberation and meaningful collaboration for next few days. Thank you very much.
Thank you, sir, for your words of gratitude. With this, we move on to felicitate our esteemed dignitaries. May I request Dr. B.C. Das, sir, and Dr. A.K. Singh, sir, to kindly felicitate Dr. W. Selvamurthy, sir, with memento and a shawl. A big round of applause, everyone. Thank you, sir. I request Dr. H.R. Chitme, sir, and Dr. Tanvi Navid, sir, to kindly felicitate Dr. B.C. Das, sir. Thank you so much, sir. I now request Dr. Vinay Lathar, sir, and Dr. Navneet Sharma, sir, to kindly felicitate Dr. A.K. Singh, sir. I request Dr. Navneet Sharma, sir, to kindly present a token to Dr. H.R. Chitme, sir. Memento presentation to Dr. H.R. Chitme, sir. I request Dr. Navneet, sir, to present a memento to Dr. Tanveer Navid, sir. A big round of applause for everyone, for every dignitary that we have with us. Thank you, everyone, that you spared your time for us. With this, ladies and gentlemen, we conclude the inaugural session of PTT DI CBR 2026. As we say, Safar abhi shuru hi hua hai, so the journey has just begun. Thank you. Thank you all to be a part of this conference. I request everybody to please stand at your place because we now have a national anthem.
भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
I request our dignitaries to kindly visit our Pharma exhibition held at Palm Court. From there, we shall proceed for the lunch. I thank everybody here for their gracious presence to attend this conference. Thank you, everyone.
Give me a thumb. मैं आके बोल Good afternoon, respected dignitaries, eminent scientists, distinguished faculty members, and honored guests and dear participants. I extend a very warm welcome to each one of you to the third International Conference on Pharma, Technology, and Translational Development, Integrating Chemistry, Bioscience, and Regulatory for Next Gen Therapeutics 2026, organized by Amity Institute of Pharmacy, Amity University, Noida. I, Lavanya, student of Amity University, is my great honor and privilege to serve you as a host for today's session. It is truly a proud moment for me to stand before you, such an esteemed gathering of globally recognized scientists, academicians, industry leaders, and researchers who are contributing towards shaping the future of pharmaceutical sciences and healthcare. With great enthusiasm, I would like to call upon stage Mr. Raghav Mudgal, the CEO of Anablade Private Limited and a sponsor for the event. I would like to call upon stage Dr. Navneet sir to present a token of appreciation. Guys, please, a huge round of applause. Thank you. We now begin with the scientific session on traditional scientific knowledge and phytopharmaceuticals, an important domain that integrates traditional medicine with the modern scientific advancements. Before we proceed with the speakers, it is my privilege and I would like to welcome our respected session chairs. First session chair for today is Dr. Anil Sharma, sir. It is my honor to welcome Dr. Anil Sharma, Vice President at Ayurveda, AML Pharmaceutics, Delhi. Dr. Sharma, sir, is a highly respected professional with an expertise extensive expertise in pharmaceutical sciences and pharmaceutical research, contributing significantly to integrating traditional medicine with modern healthcare systems. So we are honored to have you on the stage as a session chair. Secondly, I would like to call upon stage Professor Dr. Malika Pathak. It is my great pleasure to welcome Professor Dr. Malika Pathak, Professor, Department of Chemistry, Miranda House, University of Delhi. She is a distinguished academician and a researcher known for her exceptional contributions in a molecular modeling and drug interactions, and the recipient of a prestigious international recognitions. Next, for the session chair, we have Professor Dr. Harsha Kharkwal, Fellow of the Royal Society of Chemistry and Director at Amity Institute of Phytochemistry and Phytomedicine, an internationally recognized scientist with over 24 years of experience and a significant contributions in natural product chemistry, drug delivery, and pharmaceutical researches with multiple publications and patents. We are deeply honored to have such a wonderful session chair with us for the session. Now, for the speakers, the first speaker, Mr. Rajesh Arora, sir. It is my privilege to welcome Dr. Rajesh Arora, sir, for the speaker session.
May I start? Yeah, that's okay. That's okay, it's fine. I'll start.
Good afternoon, everyone. It's my privilege to welcome Dr. Rajesh Arora, Senior Scientist and Additional Director at DRDO, Government of India, a globally recognized researcher, Marie Curie Fellow, and Fellow of Royal Society of Chemistry, with over 250 publications, 17 patents, and numerous prestigious international awards, including the DRDO Scientist of the Year Award. His pioneering contribution in drug design, biotechnology, and disaster medicine have significantly advanced healthcare and scientific preparedness. Sir, it is a privilege to invite you to deliver your expert lecture. Good afternoon, sir.
Thank you. Thank you, ma'am.
respected Professor Anil Sharma, sir, Professor Malika Pathak, whom I've known for the last several years. And we've had the opportunity of sharing the stage maybe many times in Amity University itself. So it's an honor and privilege to be here. And also thanks to and greetings to Professor Kharakwal, head of the department, Amity University Institute of Pharmacy, faculty members, eminent scientists, professors, dear students. A very good afternoon to all of you. The most difficult task in this world, can you tell what it could be? Students, the most difficult task in this globe, in this universe rather, it is to keep the audience awake after a sumptuous lunch. So today I'm going to, so my endeavor will only be to keep you awake so that you can listen to what I have to share. So today I'm going to present what kind of disruptive profilotherapeutics technologies can be utilized for defense applications and some of the spin-offs for the civil society. That is where DRDO works. So basically the presentation is organized in this manner. And since the session is on traditional medicine and phytopharmaceuticals, so some research that we have done in the past, we will be sharing because now DRDO is fast moving towards state-of-the-art technologies, moving away from these phytopharmaceuticals. But over the period of several years, our cluster has made significant contributions. And Professor Sharma can vouch for it that one of the leading products of IMIL pharmaceuticals is from DRDO. So it's a great privilege to be at a place where the translational research has been carried forward by the industry. And there could be no other better person than Professor Sharma to represent that. So thank you, sir. And thanks to Navneet for giving this opportunity to be here. So there are some sustainable development goals that the UN has promulgated. And of course, traditional medicine is very important. And that is one of the focus areas that the UN has highlighted. Now, life expectancy is increasing. All of you agree with me or not? You see all old people all around the globe. Of course, India has a very young population, so that's to our advantage. But few years down the line, we will also be in the category of Japan and other nations where we would be having octogenarians and other older people. Now, globally, if life expectancy is increasing for a multitude of factors, how do you take care of the health of all these people? Now, health for all for health, and one health. These terminologies you see everywhere. They keep on changing, but it's basically old wine and new bottle. So what you have to do is that you have to take care of the blooming population, which would be somewhere like 9 billion or so in a very short time. So to cater to the health needs, the food needs of the immense population that we have within our country, we have to have revolutions. And it all started with the food revolution, thanks to Dr. M.S. Swaminathan Saab, that we have from a country that was Professor Dr. Selvamurthy often used to say that we were from a begging bowl, we have now come to a situation where food sufficiency is there and even export has been possible. Then the milk revolution came, which brought about a great transformation, and the country has been able to give nutrition to the population. Then the IT revolution, the nanotechnology revolution, the biotech revolution, and this century is now going to belong to the bio-revolution. On the other hand, we have disparity, huge disparity in health in the country. On the one hand, we have a very large population, many of which belong to the lower strata of society economically, and we have huge hospitals, which only the rich can afford. So how do you address the health needs of these diverse populations? The global population is going to rise, so we need affordable, accessible, and acceptable healthcare, and mainstreaming integrative medicine is the only option. Then on the other hand, in modern medicine, do we have a panacea? So this is a cartoon where when a person is saying that your doctor can only do so much, the rest is up to you, stop getting older. So basically, the onus then comes on the individual. What to do in such a scenario? Here's a list of some of the incurable or curable diseases, you may call it. Can anybody tell me that these diseases, do we have a cure in modern medicine for any of these diseases? Do we have? Yes, we do. Okay. Thank you for educating me, but let me tell you, there is no cure for these diseases. They are only some treatment options that are available. So the Jivan Chakra, or the wheel of life, it keeps on moving, and you need, of course, modern medicine, complementary medicine, traditional Chinese medicine, or maybe Ayurveda and other kinds of medicines, if you have to cater to the needs of the immense population, because it has to be affordable. Healthcare cannot be something that is non-affordable. So it has to be a mixture of everything. In this wheel, you can see that how different disciplines, different modalities are merging together. This I've already discussed. So the advantage of India is that we have Ayurveda, which is the most ancient Indian system of medicine, Unani and Siddha also, which can take care of many of these diseases for which there is no treatment available. Now, how many of you would agree that the medicines that we eat today, are they really chemicals, or they have their origin in plants or bacteria somewhere? Can somebody tell me, the students? Plant-based medicines. Okay, so here's a list of some of the drugs that are used in allopathic systems of medicine, in modern medicine, and they all have their origin from plants, whether they are the anti-cancer drugs, whether they are the painkillers, or you have antiviral drugs even for that matter. So the WHO has very recently made a plan for the 2035 roadmap, which the meeting took place in Delhi itself, and our Honorable Prime Minister, Mr. Narendra Modi, was there. So it was highlighted that the alternative medicine is used by nearly 80% of the population, and people have a lot of faith in the Indian systems of medicine and also traditional system of medicine world over. And it is not just because these kinds of medicines have been used for maybe 5 years or 10 years. It's a huge data on humans that is available. They have been used for the last maybe 5,000 years. And very effectively, we have been able to control, even prevent, treat, and even control the diseases. The shift in the view of the scientists and the society came about in the year 2015, because earlier the view was that modern medicine is separate, the traditional medicine, Indian medicine or traditional Chinese medicine is separate. But now with the award of the Nobel Prize in 2015, changed things a bit. Avermectin from Streptomyces avermitilis and artemisinin from Artemisia annua. So these were two very important drugs that owe their origin to plants, and two Indian laboratories. One is CDRI and Central Institute of Medicinal and Aromatic Plants. We were working in this area. I was associated with that group, and we were perhaps the first to isolate artemisinin in the world, but it was not reported due to some reasons, and we were left behind. And you can see that how the world went ahead. So here you can see that how Artemisia, CDRI, and CMAP had taken it up in a very big way, but then we were left out because translational research did not happen. With that, I moved to the diverse extreme terrain and environment because I'm from DRDO. So I would like to give a glimpse of what we do in DRDO because a lot of students are there. We have borders all around and all kinds of temperatures ranging from -55 to +55 degrees Celsius, hot, humid conditions, and nearly all environmental conditions on Earth are there in our country. And then we have the military platforms, some of which you must have seen on television during the Operation Sindhu. So we are a diverse group of laboratories in few clusters. Of course, these keep on changing, but right from missiles to armaments to soldier support system, we work in these directions. And our technologies developed indigenously. We are talking of Make in India, Made in India. So these things have come from DRDO contributions. And we are amongst the 4 nations to have multi-level strategic deterrence capabilities, to have our own ASAT, and have our own BMD program, have our own nuclear-powered submarines, even our own main battle tank, our own combat aircraft, and many things. Just a small glimpse of the activities of DRDO. And this is of course an old video, not very recent, but some of the corporate technologies that were developed for the country. And many of these and now in operation. This is Astra. And many of these products are totally indigenous product now is approximately 125 lakh. A huge amount of expenditure is being generated for the country. Besides making the country indigenously self-reliant. This is the ASAT technology whereby we can the speed of several thousand kilometers per second, and that is So soldier support technologies is just one of the clusters in DRDO, where we focus on the soldier primarily with respect to threat protection, operational support, human capital, and a large number of other things. We focus to enhance the capability of the soldier in terms of lethality, efficiency, sustainability, and survivability under all kinds of environments, extreme environments, and combat missions. For threat reduction in the area of chemical, biological, radiological, and nuclear protection, we have developed radio protectors and mitigators, which can protect our soldiers and also the civil population against some of the nuclear blasts that occur, or dirty bomb explosions, or nuclear accidents and emergencies, or even nuclear reactor meltdowns. These are some of the prophylactics that have been developed, and they are a part of the CBRN emergency kit that is given to the armed forces. Then for fortifying the combat manpower, many of these drugs have been developed and they have been given to the armed forces to protect them against the NBC incidents. Then for several of the submarines also, we have developed the technology, and these have been handed over to the services. As far as the herbal radiation countermeasures are concerned, we were involved in this research from the last several years, and Podophyllum hexandrum, Hippophyramnoids, Rhodiola imbricata, all these herbal compounds herbal origin compounds from these plants have been shown to render radiation protection and very significant radiation protection. And currently, the current status is that these compounds are undergoing trials in the non-human primates, and they will be inducted after that. Then we also isolated a radio-resistant bacteria for radio protection, and we have also isolated the individual compound, and again, that is being pursued very rigorously, undergoing non-human primate trials, and very shortly we might be having a drug from this as well. And we have characterized this compound, purified and characterized it. Then in the area of biothreat mitigation, we have a BSL-4 facility for handling of all kinds of high-risk group biothreats. And we also have a mobile point of care detection and diagnosis capability. On the right-hand side, sir, you can see Dr. R.K. Sharma, sir, also your revered batchmate. Of course, he passed away a few days back, so I would like to dedicate this to him. And molecular profiling of biothreat agents also is being done. We have a high containment facility where all kinds of the viruses and bacteria can be handled, and also a bioprocess scale-up facility for making vaccines, etc., in times of crisis in a very short time. In Air Force missions and deep space missions also, we have developed certain technologies, and on the right lower hand side, you can see a plant that is Rhodiola imbricata, on which we have worked, and this was mentioned by the Honorable Prime Minister also in Man Ki Baat. Then for naval protection also, we are engaged in technologies, and one of the plant was given to us by the former Naval Chief, Professor Admiral Nirmal Mehta ji, and we carried out studies, and the results were given to him for some purposes. Then for LIC operations, the capsicum-based grenades that has been developed from Bhoot Jalakia, which is the which is having the highest Scoville units. So the condition was this, that in one of the operations in J&K, terrorist operation, anti-terror operations in J&K, this was used, and the people, they came out running. Okay, if you want, you please kill us, but don't use this against us. Then the high altitude battlefield Siachen is the playground of the soldiers and also for us. So there are many challenges at high altitude which we address in our institute, and one of the major problems is frostbite, and we developed an allocal-based cream. I would like to just point this out to Sharma sir. Sir, we developed this cream, you would be aware of it, and the technology was given to Dabur, Sarvottam and Srinath. And there used to be some 900 to 1,000 cases around 6 years back, and many of these amputation cases just as a result of frostbite. And as a result of the use of our cream and also other modalities, from last 5 years, there has been zero amputation in the soldiers. So we wanted to transfer this technology to IMIL as well. We got in touch with some of the people, but perhaps we did not get in touch with you. So this is still available, sir. If you wish, we can give it to you from our side. So here you can see some of the pictures of treatment, that how frostbite affects and how it can only be prevented. Then for recalcitrant wound management, we have developed a cream, again, a herbal-based, hippophy-based cream. And this, of course, we have given to you also, sir. And IMIL has manufactured this. And this is the star product from Diber, earlier DARL, formerly it used to be called as DARL, Leucoskin, which is very useful in leukoderma patients. And sir, has been involved in this work. And thanks to you, sir, that this technology could reach the market and help so many people. Then there's a dip GFIT that we have developed from Ganoderma lucidum. This is also undergoing clinical trials, and we would be transferring the technology to few of the companies, and we welcome any of the pharma companies who would like to come and take this technology. Then a herbal immunological and adjuvant has also been developed and given to the industry for jungle warfare protection, especially snake repellents. Many things have been developed which are primarily herbal in origin. And for disease vector control, also a large number of technologies have been developed by DRL Tejpur. So some of the emerging trends that are going to shape defense technology in future include biochips, on which we are working. One of the biochip was recently in the news, wherein we have collaborated with Delhi University to develop, which will predict early heart attack incidents in soldiers much ahead of the attack that takes place. Then big data is one area where the focus is there, then quantum communication, and of course, robotics and AI. From operational stress to predictive profilotherapeutics, we are fast moving, and it's a systems defense biology framework. Some of the areas we are focusing to some extent, but we are again moving from these areas to more advanced areas. Then big data is the new strategic high ground, and we are engaging ourselves in this now. And also digital twins, etc. This kind of research is being carried out. So with this, I would like to thank my organization, my director, and also to Dr. Navneet, the entire team at Amity, and the chairpersons. Thank you very much. And to all of you, thanks for a very patient hearing. I hope, and I'm very happy to see that none of you slept. So my task is complete. Thank you. Thank you.
Thank you very much, sir. It's one of the very key area what you have highlighted. So not merely DRDO is not merely for the defense of the soldiers, but it is for the defense of the society as well. They have very deep interaction with the villagers also and the people who are located in various tribal areas. As he has mentioned, we have the technological transfer collaborations with DRDO, and thereby we were interacting for the last 2 decades, more than 2 decades, with the DRDO on various occasions. Some of the field, what he has not highlighted, particularly I remember the days of the corona pandemics, the way DPAS particularly had contributed during that time. I feel we all should salute DRDO and the eminent scientists, the way they have diagnosed the people, and so quickly in the Palam area, they developed the hospital and the provisions for the hospitalization. And that time, why I know that, because that time they gave us the technology to manufacture sanitizers. So it was again a formulation what was given by them, and that too, that formulation was free of cost and free of the royalty. So in the interest of the society, what DRDO has given during that time, we must acknowledge with all big gratitude.
Thank you, sir.
We are very honored at Emil Pharmaceuticals, particularly for the Leucoskin. At your institute only we initiated in the year 2003, when they offered the pharma companies for the technology transfer. We opted for some skin preparations. And leukoderma, particularly, our chairman sir was very much interested in leukoderma, though it is not a disease condition. Everybody is looking for the medicine, right? Even the pharma company is looking for the medicines. But what about the social stigma, which itself is a very big psychological disease? So we had a couple of the people in our surrounding which were suffering from the vitiligo, and that's why the company had the interest. So that time in the year 2003 and 4 only, we had taken the technology from the Diber. That time it was in Pithoragarh. And we happened to coordinate with the DRDO headquarters also. That time it used to be Sena Bhavan. And we came to know that not merely the defense factor, but many of the technological innovation is there from the DRDO, including the helmet. So helmet was developed by them. The concept of the packed food. That time the Brigadier Umang Kapoorji was heading DRDO that time on the technological part. So I had interacted with him. So how the food is to be packed and how it is to be dropped to the army area through the helicopter. So he was just telling the story that how the concept of the packed food was developed by them. That also is very, very important. So they have a big venture for the health industry. And many of the, I know few of the scientists who are very deeply working into the field of the pharma sector and health industry. Even your lab in Gwalior also is very, very effective. And once again, on behalf of all pharma industry and our institute, we thank you very much, sir.
Thank you.
Big regards to DRDO. And I wish that let's have a round of applause for DRDO. Thank you.
Thank you very much, sir. The slides that I had removed just before coming, sir, you have highlighted, so many thanks to you for that, sir, because I had included everything. Yes, sir, very well pointed out that during COVID, we worked almost 24/7, and we had a laboratory set up in 2 days' time for diagnosis of. We were the first to set up in the entire NCR region, or rather in the country itself. Within 2 days, we set it up. And ours was a WHO recognized and ICMR recognized laboratory. And we did the testing free for the entire population. Whosoever came, we did it, risking our lives. And many of us fell prey to COVID. Many of us, unfortunately, some of our very dear colleagues passed away due to that. And even I am alive because we had set up a hospital and I spent 21 days in ICU and returned. I am standing before you because we were able to have the hospital set up in a very short time when people were dying from paucity of oxygen. Then sir also highlighted about the food technologies. So the MTR food, the Haldiram food, the ITC food, whatever packages you see, it is the technology that was developed by DRDO. This was initially started as a result of the result of the space exploration. So when Wing Commander Rakesh Sharma went into space, the food was supplied to him, and the entire food was made by DFRL Mysore, our DRDO laboratory, and the technology was subsequently given to the industry. So there are a huge number of spin-offs. In the Indian Railways, if you travel now, those of us who have traveled maybe 15, 20 years back would know the kind of stench we felt on railway platforms or when we were traveling by train. So the biodigester technology, again, it's a technology wherein the bacteria were brought from Antarctica and Siachen Glacier and developed into a consortium and introduced. And then we worked with the Indian Railways to have that technology. And now almost all bogies of the Indian Railways are fitted with that technology. So it's a huge contribution to the civil sector also that we have made. Of course, in the border areas also, we do a lot of work in very remote areas. And even for Gaganyaan project, we have been contributing. The food is again going from TFRL. And there are many other technologies, several pharmaceutical technologies also from INMAS, that are a part of the Gaganyaan, and even from DIPAS, we are contributing in a big way. So thank you very much, sir, for your comments and this, and all the chairpersons. Thanks a lot. Thank you very much.
I kindly request Dr. Anil Sharma, sir, Dr. Malika Pathak, ma'am, and Dr. Harsha Kharkwal, ma'am, to felicitate our first speaker for today, Dr. Rajesh Arora, sir.
स्क्रीन पर भी एमिटी का फोटो भेज बेटा.
Thank you, sir, for sharing such a wonderful presentation. Yes, sir. Yes, sir. Thank you.
We move on to the next speaker for today. I would like to request the session chairs to introduce the second speaker for today.
So it is my honor to introduce Dr. Gandhi. It is my honor to introduce Dr. Galib, Additional Professor at the All India Institute of Ayurveda. Dr. Galib is an eminent researcher with over 80 publications, significant international citations, and prestigious awards, including the Sheikh Zayed International Award. His contribution to Ayurvedic pharmaceutics and traditional medicine research are highly respected globally. Sir, we welcome you to share your valuable insights. Welcome, sir.
Okay. Yep. Yeah, thank you so much. A very good evening. The respected chair, Dr. Harsha ji and other panel members, senior faculty members, dear students. Possible that there may be some faculty members from the Amity University. And a very good evening. And happy to see what Rajesh ji, okay, so he was presenting over here. And I'm touched with that tagline. So what is mentioned as Balasya Moolam Vignanam. So which is the tagline of DRDO. So which says that, yes, the science is the power which propels us, which keeps us standing in the society. So thank you so much for enlightening us with your experienced scientific journey. And even I'm happy to know that you are the first, otherwise, so you are the very preliminary individuals as seniors, scientists, okay, so who have worked upon artemisinin. We know that artemisinin is with the name of UU only, but okay, so we came to know that, okay, so you are the initial people, okay, so who have worked with the Artemisia annua. So thank you so much for your wonderful presentation. And I am not, hardly, okay, so I will be addressing to the students who are sitting there in the back benches. They are the backbones of tomorrow's society. The thing is when, of course, because I am from the traditional field. I am from Ayurveda. I work for Ayurveda, All India Institute of Ayurveda. Whenever, okay, so you speak something about traditional systems, you suppose, and when you search for what is that traditional system of medicine, what is that Ayurveda? So some of the websites, okay, so they says that it's a pseudoscience. And I would like to say that, okay, so whether it is a few of the slides, okay, so I will say that what exactly, what kind of scientific background, okay, so it has. And as a student, as a scientist, okay, so where we can tap that potential, what kind of potential we have. Of course, okay, so rightly mentioned by Rajesh ji, okay, so let it be communicable disease, let it be non-communicable disease. So there may be a battery of pathological entities, okay, so that has been shown in one of the slides. So for which the treatment is there or not, okay, so treatment is something different, okay, so whether management is there or not, whether we can improve the quality of life of those individuals who are suffering with those pathologies or not, okay, so there are a huge number of questions. That may be one of the reason why the global healthcare, okay, so they are looking towards the traditional systems of medicines where I'm not saying that, okay, so we can provide all those answers for which the people, otherwise the science is looking for, but there are certain areas where the scientists can tap that potential. So there is a scope that we can see it. So in brief, in precise, okay, so I'll be touching, okay, so these areas, what are those, basically, what are those areas where, okay, so product development related information is available in the traditional systems that I will, and what kind of opportunities that I will focus much upon. Everyone knows it very well, okay, so how this traditional system has been, okay, so traditional science has been, traditional healthcare has been defined in the WHO definition. So you can say that, okay, so they said that, okay, so it is nature-based remedies, okay, so it has been, and then they said that it may be codified, it may be non-codified, and okay, so they focus as much upon the, okay, personalized approach and all those things, okay, so it has been very clearly mentioned in that definition, but rightly mentioned by the earlier speaker. This is the third global survey, which has been very recently, which has been released in the second global summit at Pragati Maidan, okay, so in the month of December 2025. Okay, so they say that, okay, yes, the demand for the traditional systems, it is increasing across the globe. For the traditional, okay, so systems of medicines, it is increasing, popularity is increasing, demand is increasing across the globe, and these digits, okay, so they clearly says that, yes, okay, so there is demand, not only in Indian scenario, but across the globe, okay, so that demand is increasing. At the same time, okay, so if you think that why the demand is increasing, so why the traditional systems, why that global pendulum, global healthcare system is drastically moving towards the traditional systems of medicines. So one of that editorial, if at all you are looking to support that perspective, if at all you are looking to suppose, they have enlisted why it is so. Right, so it may be adversities which is related to the with the conventional system of medicine. It may be, right, compromised QOL. Okay, so they may be managing a system, they may be managing a pathology, they may be managing some symptoms of that specific pathology, but at the same time, okay, so there are battery of other reactions that are entering, the battery of other manifestations that are observing in that management period. So it is compromised the quality of life of the individual. So that may be another reason. And then therapeutic approaches, that's what, okay, so sir was asking for diabetes, whether you have cure, whether for the hypertension, whether you have cure for obesity, for polycystic, okay, ovarian syndrome. So do you have cure for all those diseases? So unfortunately, no. So we can't say that, yes, there is treatment. We can't say that. So we can improve the quality of life. We can manage the symptoms to some extent at this moment. So 100% cure, 100% treatment is not available for these many of the diseases. And another thing is, okay, so the drug resistance or antimicrobial resistance or drug dependency, okay, so that is being experienced at multiple levels. Okay, so that is also one reason which is mentioned in that perspective. and that is experienced worldwide. Okay, so Lancet's one of the editorial which was published way back in 2000, they said that antimicrobial resistance and the global antimicrobial agents, okay, so they are running out of the pocket. Okay, so this is what it has been referred, and that may be one of the reason why the healthcare professionals, scientists, they are moving. Okay, so let it be whatever it may be the stream, it may be ethnobotany or pharmacognosy or pharmacology or plant ecology. People, they started to data mining, so they started to search, okay, so what kind of molecules are otherwise, okay, so what kind of information that is available in the traditional healthcare systems, and how we can tap that specific area and how we can develop new molecules. So in that scenario, so if at all we are coming, you suppose these traditional systems in Indian scenario, so it is popularly known as Ayush, which stands for, okay, so all those what I can say is, okay, so India, I feel, is one country, I feel, okay, so it is only one country across the globe which has legalized these, all those, okay, so medical traditional systems. Okay, so India is the one country which is enjoying that legality for, okay, medical pluralism is existing. Not only the conventional system of medicine, but even, okay, so Ayush, Ayurveda is legalized, Siddha is legalized, homeopathy is legalized, Unani is legalized. So that legal entity is not available in many of the countries other than India. So this is one beautiful country, okay, so where we have that legality. So at the currently, okay, so this Ayush, it is being managed by Ministry of Ayush Systems. Of course, okay, so the details are there in that one, but what I can say is currently we are in a very receptive environment. Ayush is in a receptive environment where encouragement is being noticed at multiple platforms. You can see that, okay, so starting from our Honorable Prime Minister to the latest, that second global summit in in New Delhi. So you can see Man Ki Baat. Okay, so there are more than 140 episodes of Man Ki Baat. If at all we are analyzing this. Suppose there are more than 48 times. Okay, so Honorable Prime Minister, he took the reference of traditional systems of medicines. He may not be talking exclusively about Ayurveda or Siddha or yoga, but okay, so he's taking the reference of traditional systems of medicines, and he want to uplift the systems. So it itself indicates that, yes, okay, so when the first man of the country, if at all he is, okay, so focusing much upon that, you suppose, okay, so what else is needed? So we are in a very receptive environment at this moment. Not only that G20, but let it be T20, let it be Y20, let it be W20, okay, right? So in each and every scientific platforms, the traditional systems are being discussed. So it became, traditional systems of medicine became a routine agenda item to be discussed in each and every forums. Okay, so in this couple of years. So let it be ICD-11 or, okay, so integrative health is, it's not a new for us at this moment, because already, okay, so huge amount of work, okay, so it has been initiated. So let it be, okay, so few examples, okay, so I was putting over there, okay, so let it be AIIMS Delhi or Jodhpur or Nagpur or Rishikesh, okay, so they have already initiated projects with, by joining hands with the traditional healers, let it be Ayush or CCRS or whatever it may be, okay, so they have already initiated. So they are trying to come out to generate certain evidences to the age-old remedies. So this is just to say that, yes, receptive environment is there, but if at all you are looking towards the other facet of the coin, you suppose what the consumer is feeling even today in 2025, okay, so when we are standing, okay, so the consumer feels that, okay, so they are safe, they don't need any scientific evidences, they can be used by procuring the products over the counter, okay, so they doesn't have, okay, right, the expiry date, right? So they can be used long. Okay, so even this perception is also there. Okay, so once if it is prescribed, you suppose, okay, so that can be used for years together. Okay, so those limitations are not there. Okay, so they can be used as and when, okay, so it is needed. Okay, so such kind of perceptions are there, but they are only the perceptions. What I prefer to say is, okay, so though they are the molecules, though they are the formulations which are coming from the nature, which have been manufactured, which have been prepared, okay, so those formulations by using the natural resources, but they are the medicines. So they are supposed to be considered as medicines. They are supposed to be used under the supervision of a qualified registered Ayush practitioners, and all those do's and don'ts, okay, so which are which are supposed to be followed even in case of conventional systems is equally applicable to the traditional systems of medicines. And a few of the right concepts, okay, so I would like to put it over here that, okay, so while administering a medicine, okay, so they say, the classic says very clearly that, okay, so one has to be very careful. It's not that, okay, so all the traditional molecules are safe. Even there is a possibility of adverse drug reactions by using traditional systems of medicines. Adversity is not far away, okay, so far away from the traditional systems. It's not so. Of course, okay, so the adverse drug reactions in relation to the conventional molecules, it may be lesser. They may be mild, they may be moderate, but they may not be life-threatening. They may not be fatal, but there is a possibility of adverse drug reactions. Okay, so that's why one need to be very careful. That is what, okay, so classical context, okay, so in that classical context, the literature says that one has to be very careful while using any sort of medicine, while using the traditional molecule, okay, so this is what they are saying. They says that, okay, so how the dose is supposed to be tapered, okay, so what is the dose which is useful to me? If at all it is a paracetamol, if suppose you have that, okay, so that posological concept that if at all it is used more than 2 grams in 24 hours, it may create hepatotoxicity. So similar kind of right concept, they have been mentioned in the classical literature that, okay, so you have to observe the, okay, so various factors which are responsible for the therapeutic right efficacy. Looking into those factors, okay, so one has to taper the dose, one has to increase the dose, okay, so one has to okay, right, adjust the dose depending upon the okay factors, okay, so it has been very clearly, it is mentioned there. And continuous administration, this happens many a times. Once if the medicine is prescribed, okay, so they will use it for years together without taking the right consent of that physician, years together, okay, so many cases, okay, so we have noticed in recent past that, okay, so once it is prescribed almost in 2000, for example, 2020, 25, okay, so since last 24 years, they are using the same medicine. And they come with some sort of inconvenience that, okay, so this kind of new manifestations are being noticed. So there is no supervision. So without supervision, medicines, if at all they are, okay, so they may be any sort of medicine, they may be Ayurveda medicine, it may be Siddha medicine, it may be Unani medicine, it may be homeopathy medicine, but regular visits to the qualified physician is very much essential. It doesn't mean, okay, so this is very clearly, it is mentioned in the classical literature that you should consult your physician at regular intervals. So this is very clearly mentioned. So similarly, minute dosing, okay, so right, so we know that, CS know that, the classics know that, okay, so as rightly mentioned by right Rajesh, right, so classic, it belongs to 5,000 years back. They knows that, okay, so it contains some sort of element which can create some sort of adversity. That's why they said that, okay, so it has to be used in the dose of 1 milligram. They knows that. They knows that it creates toxicity, it creates adversity, it creates some sort of inconvenience if that understanding is not there. It has been very clearly mentioned. That's why, okay, so you can see that, okay, so the classic says that you have to start the dose from 1 milligram. You have to start the dosage from 1 milligram. So it means that, okay, so they have that understanding of PKPD, kinetics and dynamics, and how the body will metabolize, okay, so where it is going to be metabolized and how it will be created. And then, okay, so what kind of toxicity can be anticipated, okay, so that beautiful, information, they have it. So if at all it is improperly processed, what happens? If at all the drug doesn't have that quality, what happens? Okay, so all that information, it is very clearly, it is mentioned there, okay, so in that in the classical literature. So that's why I say that it's not a pseudoscience. So it is based upon, okay, so its own principles. So all those principles, of course, okay, so it has been written in Sanskrit. It doesn't mean that it doesn't have any value. Fine. The only thing is, okay, so we need to reframe that information into specific, right? So we need to speak out, okay, so that information in a language where the global science can understand it. Okay, so that is the what, okay, so that is the scenario which is needed now. And if at all you are looking, you suppose, okay, so into that classical literature, of course, we have huge amount of those dosage forms, let it be solid dosage forms or semi-solid dosage forms, powders, tablets, capsules, gels, right? Topical dosage forms and dusting powders. And so all those dosage forms, and for all those dosage forms, okay, so we have SOPs, standard operating procedures. Okay, so if at all it is topical application, so let it be, okay, so whatever it may be a paste, if at all you want to prepare for a topical application, how that paste is supposed to be prepared, what are those okay, so quality standards which are essential to be complied before it is coming to the clinical setup. Okay, so all those things, okay, so which have been very clearly mentioned. For example, okay, so I'll put this Of course, it is there in a classical, right, in a Sanskrit verse it is. But so if at all you are preparing a liquid dosage form, you suppose, okay, so what are those operative guidelines which are supposed to be considered? What are those endpoints where, okay, so you have to stop that, okay, so preparation of that specific fine preparation, and what are those post-operative precautions you need to take it? Okay, so whether it has that life or not, okay, so whether it retains that shelf life or not, whether it is okay effective to be launched into the market or not. Okay, so these guidelines they have been very clearly they have mentioned, and there are many such kind of verses which are available there. Okay, so just one example I'm quoting over there. Okay, so you may be knowing that, okay, so this is a schedule even drug, right? Schedule even aconitum ferox, aconitum jasminum, whatever it may be. Okay, so it contains aconitin, which is lethal molecule, right? So they know say that, okay, so this is cardiotoxic element. Okay, so they knows that, okay, so whenever you are using this specific herb, you need to be very careful. You need to be very careful. Okay, so whenever you are using this in a formulation, that cannot be used, right, without, okay, so having any qualified, right, professionals advice. They says that, okay, so they said that it is hrit gati rodhanam mrityum cha. It may even create otherwise, okay, so it can become fatal if at all you are using, okay, so inappropriately. So it has been very clearly mentioned where the dose is supposed to be started and how that dose is supposed to be tapered if at all you are, okay, coming across with some sort of inconveniences. Okay, so all those things they have been very clearly mentioned. This is just one example. Similarly, okay, so they have given examples for all those schedule even drugs, all those metals and minerals. Okay, so you know that, okay, so we are using mercury, right? So how mercury is supposed to be used, how mercury is supposed to be processed, how the mercury, that toxic mercury can be converted into therapeutically viable molecule. So all those things very beautifully, okay, so they have been explained over there. And I would like to draw your attention here, right? So this is very one verse, okay, so which says that, yes, we have all those quality standards. Okay, so you can see that, right? So all those good agriculture practices, how a product is supposed to be developed, if at all, okay, phases of various product development, right? Okay, so we have to start from the collection, right? So product, the sample collection and sample storage, where it is supposed to be stored, how it is supposed to be stored, what are those good qualities of a storing chamber, fine. Okay, so good storing practices and how the drug is supposed to be analyzed, whatever you are doing in pharmacognosy, right? Powder microscopy and I don't know. Okay, so all those, microscopy, right, and histology and whatever you are doing. Okay, so how the product is supposed to be, how the raw material is supposed to be identified, and how it is supposed to be. Okay, so what kind of name it is supposed to be given to it? What are those posological considerations? Fine. What is that GMP? Good manufacturing practices. Fine. So good laboratory practices, good clinical practices. Okay, so all those things that, okay, so starting from the raw material collection to product development to okay, so it's preclinical evaluation, fine, it's toxicity evaluation, then, okay, so phase wise, right, a clinical evaluation. What they are saying is pranishusthapya nirdesham manavam prati. Okay, so how that efficacy is supposed to be established and evaluated. So they have been, beautifully explained in that one specific verse. It doesn't mean कि, right, so classical literature में GMP नहीं है, classical literature में quality control नहीं है. It's not like that. The thing is, okay, so they have mentioned it in their own way. Now we are in 2020, we are in 21st century. So we have, if at all I want to convince the global community, suppose I need to speak in their terms. I need to speak, okay, so in their language. So that is the only thing, okay, so which is lacking, I feel. Okay, right, so you can see that, okay, so the same shloka, the same verse, if at all you are breaking it into 3 different phases, you suppose, okay, so it has that scientific definition which you, right, follows. So it has that proof of identity and purity. And then, okay, so it has that content check. All those 3 different phases, okay, so which comes in that process of drug development, all those things, okay, so they have mentioned in these 7 or 8 lines. Fine, it is there. That's what, okay, so you used to product development when it comes, okay, so you used to see all those things, okay, so you used to see the passport data, you used to see that whether the product is authentic or not, whether it is the same species or not, which I am collecting and which I am exercising. Fine. Okay, so you used to see, okay, so all those organoleptic characters, you used to evaluate that biological activity in preclinical, otherwise cell line models. Fine. After that, okay, so you will evaluate it for scientific verification that yes, phase 1, phase 2, phase 3, phase 4, post-marketing surveillance, and all those things you will do it. So that is what, okay, so it has been mentioned over there. So in nutshell, okay, so what I would like to tell is, okay, yes, okay, so all those concepts which we are following in the process of drug development, yes, okay, so we are following it. We are even, okay, so today, of course, we are following it, but it has, okay, so beautifully, okay, so it has been explained in the classical literature. So all those regulations, okay, so they are even applicable to the traditional systems. Let it be, right, Drugs and Cosmetics Act and, right, so we have that, okay, so our education, training, registration, and all those things, okay, though they are well regulated, we have all those guidelines, okay, so which are existing, okay, so in this moment. And as I said, okay, so if at all some sort of inconvenience is there by using, okay, so those formulations, you suppose, of course, pharmacovigilance is also, right, the system it is existing, okay, so in the scenario. But the thing is, okay, so of course, system is existing, traditional knowledge is there, legality is there, everything is there. What the this decade document of WHO, what they're saying is, yes, besides that, right, integration, fine, okay, so they have those 4 bullet points, okay, so which have been mentioned in that specific document, okay, so Global Traditional Medicine Strategy 2534, okay, so they say that, okay, so collaborative and integrative approaches, okay, so they have been very clearly mentioned in that specific document. This says that, yes, okay, so traditional knowledge, okay, so is one potential area, so which can be useful, which can be tapped for the global healthcare, okay, so that is one. And if at all you want to generate the evidence, you suppose, okay, so we need to work in a collaborative mode, okay, so this is what, okay, so they have mentioned. Of course, okay, so in that domain, okay, so many, of course, okay, so this is mentioned by Rajesh ji just now. And many isolated molecules, they have been invented, okay, from Raulfia to, right, artemisinin, okay, so many huge amount of molecules, they have been identified various for various disease entities. Huge amount of information, of course, okay, so it is available, okay, of course, I'm leaving that link over here, okay, so you can search for that. So huge amount of work, it has already been done. But of course, okay, so besides that, okay, so I can say that huge amount of information, it is already available in the classical literature where we can, right, definitely work in a collaborative mode. So just one verse, okay, so I'm putting over there, okay, so this is from Charaka Samhita, okay, so where, okay, so they speak about 500 different categories of otherwise compounds, otherwise mean different what I can say is groups of medicines that I can say. Fine, you can see that, okay, so where we can tap, okay, so how we can do it, okay, so we can work in the areas of neurocognition, we can work in the areas of, right, anti-aging, right, it may be antioxidant or it may be adaptogenic, it may be cardioprotective, it may be neuroprotective, it may be nephroprotective, it may be, right, so antimicrobial, okay, so one biggest threat, okay, so that the global scientific society is looking for, okay, so antimicrobial resistance, okay, so we can see, right, so there are enormous, okay, so information that is available there. Fine. So just this is one glimpse, okay, so from Charaka Samhita 4th chapter, okay, so it is, okay, so you can see, okay, so health supplements we can, right, so we can come back with probiotics and prebiotics, we can come back, we can work something about, okay, so antipsychotic drugs. So topical applications, if at all somebody is interested, you suppose, okay, so pruritus and psoriasis, right, so there are a huge amount of information, it is available there, okay, so we can tap that specific. And you may be interested in nutraceuticals and, right, so you may not be interested in, right, therapeutics, otherwise, what I can say, therapeutic products or therapeutic diets. Therapeutic diets is one biggest area, so where you can tap that potential. So whether I can come out with some, okay, so module, otherwise, okay, so some package for hypertension individuals, can I come out, okay, so with some therapeutic diets for a diabetic professionals, okay, so can I come out with some sort of, okay, so diabetic, sorry, food packages for the obesity individuals, okay, so therapeutic diets is one potential area, so where, okay, so we can work, okay, so therapeutic products, so developing evidences is a different thing, but okay, so cosmetics is one potential, cosmeceuticals is one potential area, so where you can work out to come out with some sort of herbal, right, skincare products and body care products. Of course, the industry is okay, it is already working in that specific domain. The society is filled with okay, so the market is filled with, okay, so various such kind of products, but still, okay, so there are many more avenues. Okay, so you can come out with, okay, so products for sun protection, what sir was mentioning about. You can come out with hair care products. You can come out with oral hygiene products. There are many nutraceuticals, and then of course, okay, so Ayurveda Ahara is one specific gazette, okay, so which has came out very recently, almost a couple of years back. That gazette came out. Okay, so under that specific gazette, okay, so you can come out with some sort of nutraceuticals, health supplements, and that is okay. So one specific, okay, so one just if at all you are interested in working a clinical domain, you suppose, okay, so you can see that, okay, so one undergoing a clinical study, okay, so I was giving credit to that department of general medicine. Okay, so they are working on some customized treatment approaches for psoriasis, whether we can have treatment for psoriasis. That's what the question which has been raised by Rajesh. You can see that I'm not saying that, okay, right, so we are treating psoriasis. It depends upon various other factors whether we can cure it or not, but at least the patient is happy now. We can see a smile on his face. We can bring, right, smile, we can bring happiness in the families. That is enough. Okay, so we may not be able to cure the pathology 100%. We may not be, but okay, right, so the patients, they are happy. Individuals, okay, so they are happy. This is one case of psoriasis that I am showing it to you. You can see the lesions before treatment and after treatment. There is a huge potential. But the thing is, again, okay, so if phytopharmaceuticals is not entirely Ayurveda medicine. Ayurveda medicine is something different. Traditional medicine is something different than the phytopharmaceuticals. Okay, so you can see that one ring, one shoe, it won't fit to everyone, right? So that's why, okay, so definition of WHO's definition, traditional medicine's definition, if at all you're looking, you suppose they said that it is a customized medicine. It is customized medicine. So if at all it's a pyrexia, you suppose, okay, paracetamol may be working for everyone, but traditional medicine, it won't work. If at all it is amla, if at all I am giving for pyrexia, I suppose that amla, it won't work for every instance of pyrexia. So that's why, okay, so we have to customize the medicine. So that is one biggest challenge, I can say. I cannot say that it's a threat, but it's a biggest challenge for traditional systems of medicines when evidences are supposed to be generated. So that's why there are unique therapeutic approaches are there in traditional systems of medicines. So that is one biggest thing, okay, so which comes in between phytopharmaceuticals and these traditional systems of medicine. And those complexities are supposed to be addressed. And then when integration comes, you suppose one big challenge will come is interactions. Herb drug interactions is one potential. That's what the recent ICMR's document, which has been released almost 6 months back. Okay, so they are coming out with RIM, Research in Integrative Medicine, is one policy which is coming up. I feel, okay, so it is not yet released, but they are coming up with that policy, which it was floated almost 6 months back for suggestions from the experts. So in that specific document of around 4 or 5 pages, okay, so they have referred about the possibility of herb drug interactions. So if suppose, okay, so I'm a diabetic, and if at all I am metformin, you suppose, or if at all I am taking insulin, you suppose, and as a traditional healer, okay, so somebody added some molecule, amla, okay, haldi, okay, so it has been prescribed, for example. So what haldi is going to do with this metformin, if at all these two are consumed at a one single time concomitantly, if at all we are using, you suppose, okay, so whether they are, okay, so what kind of interactions are anticipated? So that is one biggest area which is needed to be, okay, so that area is untouched as of now. What are those herb drug interactions? What are those, okay, so concomitant use, if at all, okay, so the individual is practicing, you suppose, then, okay, so how that area is supposed to be addressed, okay, so that is one biggest challenge. So that is what, okay, so I would like to say, okay, so this is from the Mahakavi Kalidasa's verse, okay, so from the Malavika Agnimitram. Okay, what he said is Purana Mityevana Sadhu Sarvam, right? So all that old is not good, right? All that new is not necessarily bad. The thing is, okay, so individual as a professional scientific individual, okay, so you have to analyze, okay, what is that specific theory and what kind of goodness is there, what kind of badness is there. So that you have to analyze, and then, okay, so that that information is supposed to be used, and it has to be used for the betterment of the healthcare society. Okay, so this is what he said, Mahakavi Kalidasa, he says that. So that I would like to put in front of you, and I would like to say that yes, Ayush, definitely it has immense potential in prevention and management of, I'm not saying that, okay, so curing, okay, so prevention and management of various pathologies. And of course, okay, so ICMR possible that, okay, so considering this, okay, so they are approaching they are inviting projects from multiple stakeholders. So ethical requirements, it is there already. ICMR is calling, and IKS, India Knowledge Systems, of course. So this is closed now. They have invited projects until January 30. So it is closed now, but systems are there. Let it be CSIR, let it be DBT or DST or ICMR. Okay, people, they are trying to accommodate you. Okay, so they want to, okay, so evaluate efficacies and potential of traditional systems, so which can, contribute to the global healthcare. So with that, so thank you so much for giving me this space. Thanks to Navneet and thanks to the
Thank
chair
you.
and
Thank you very much,
other
Dr.
members.
Khalid, sir.
Thank you so much.
You have very well elaborated all the perspective and scope of the Ayush system and how evidence-based we can substantiate that. That is very, very important. And I hope that we all have taken the benefit for all your vital presentation. Thank you.
Thank you
Thanks
so much.
again.
Thank you so
for
much,
your
sir.
note.
Thank you, sir, for your valuable session. I would like request Dr. Anil Sharma, sir, for facilitating our second speaker for today, Dr. Galib, sir. Dr. Galib, sir, I'll request you to come on stage, please. Dr. Harsha Kharkwal, ma'am, and Dr. Malika Pathak, ma'am, can also join.
Thank you, sir.
I would like to call upon stage Professor Yogesh Alawat, sir, Professor of SGT University, Gurugram, as a special guest for today. I kindly request the session chairs to facilitate our Professor Yogesh Alawat, sir, today. We move forward for the third speaker for today, Dr. Sujata Mohanty. I would like to request Dr. Anil Sharma, sir, for introducing her.
Thank you. Thank you very much. Now, the next session for the day is from Dr. Sujata Mohante ji. It's our great privilege to introduce Dr. Sujata Mohante ji, professor at AIIMS, New Delhi. Dr. Mohante is a globally renowned stem cell scientist, founder president of the Indian Society for Extracellular Vesicles, and a pioneer in stem cell research and regenerative medicines. With over 280 research publications, multiple international fellowships, and several prestigious awards, including the AIIMS Excellence Award and fellowships from NIH, ICMR, and international stem cell societies. Her work has significantly advanced translational and clinical stem cell research. Ma'am, we are honored to have you with us, and we invite you for your presentation. Thank you.
Very good evening to everyone. Thank you so much, Amity. Thank you so much, the organizers, and also Dr. Navneet for giving me this opportunity to come here. This is my first time visit in terms of presentation. Thank you, Chairperson, so much for your kind words. So I think I am feeling odd woman out here. I'm not a chemist, not a pharmacist, but my area of research, as said, is stem cells, which maybe comes under new investigational drug. So before that, my presentation will be the journey which our team has gone through in making stem cells and stem cells derived product for the benefit of mankind. So it is nothing extra. It is whatever what we have done, I want to present you so that you learn a little bit about the biology and how we have uh, taken care of different milestones. So I come from AIMS. So it was established in 1956 with a special act of parliament. And it has been since its inception a #1 medical hospital and also a medical research center in India, and also a very high ranking. Recently, it was #6 in medical throughout the world. So what is the mission of this AIMS? So if you see, when it was established by Dr. Rajkumari Amrit Kaur, who was the health minister and the first president of AIMS, it was to give higher education to MBBS students. That time they used to go out for MD and other DM studies. So the main aim was to give them super specialization education. So first is education, second is to research. India is a vast country having multiple diseases and the problem, which are very unique to our subcontinent. So it was mandatory that we do our medical research and find out the answers of our own special diseases, which is second, and third was the patient care. But somehow during our course of time, the patient care has taken the first. So keeping the mandate of new innovations and frontiers in research, AIMS also established Center of Excellence for Stem Cell Research, which was very well supported by Department of Biotechnology for more than 12 years. So here we are doing stem cells, which is apart from the cancer treatment, the other application of stem cells in degenerative disease. So if we think about stem cells, it comes to our mind that it is a powerhouse of regeneration, fine. And it is also, as per the DCGI and CDSCO, is a new investigational drug because it is not yet proven. It is still in a very clinical and a trial mode. So what are the AIMS experience? I will just point out few one. So we started knowing about the basic research in different areas of stem cells. So few I have listed here. And then we moved from bench, whatever we learned during our basic research, to the bedside. The first example is ocular surface reconstruction here. I think pointer is working, yes. So basically what happens in the cornea? So how do we see? Because cornea is very transparent. From there, the light enters and goes into the retina, and there is a formation of image. So what happens if your cornea is damaged? So light will not pass and there will not be any image formation and the person will be blind. So there were a lot of these type of patients in our RP center, which is a specialized center for eye care. And we thought that nature has given us some stem cells in our body. Of course, we have limbal stem cells, which are present in the eye. So why not harvest these limbal stem cells? So what are these stem cells in the eye? We blink our eyes. So every time we blink, there's epithelial cells on the topmost layer of our cornea, which is shed off. So there has to be a continuous supply of these epithelial cells to keep your cornea transparent and also moisturized. So this is the limbus area which I was talking about between the black and the white of your eyes. From here, we take the tissue and grow these limbal epithelial stem cells. So this is another way to show you what is the limbus between the white and the black. And there is a pool of these limbal epithelial stem cells which migrate as per the need towards the cornea of our eye. So for that, what we did, Shweta was the girl. She tried to do some of the basic work. This is the limbus which we got. Initially, we started with the cadaver. Then we got it from the donor. This is the small piece of the limbus from the donor eye. If other eyes is unaffected, you grow them in the lab and you try to see that how you can turn this eye into a good eye. So we need a scaffold to grow these cells and how to transplant these cells because cells are very fragile. So we use amniotic membrane, which is again a very age-old practice, a good source of stem cells and anti-inflammatory. On these amniotic membrane as a scaffold, we grow these limbal epithelial stem cells. So Dr. Radhika Tandon is the surgeon who is collaborating with us. So you can see here, this is the unaffected eye. From here, small piece of biopsy is taken. which is at the tip of this forceps, you can see here. And then it is given back to the lab. We grow them in the lab for around 14 days at the ambient temperature with proper media, and then full characterization before we get some ethical approval. I will not go into this detail. So this is the affected eye. So you first remove this fibrostic tissue, and then you transplant that amniotic membrane which contains epithelial stem cells. And then either it is sutured or contact lens bandages put there on the eye. So this is before. This is a 17-year-old girl which is having a chemical injury. So this work started because in our OPD, we saw a lot of young people, like very young, 15, 16, 12, they are working in the factories and there is no protective gear for them. And there's a lot of hazardous material and they are almost blind because of those injury. So you can see after a limbal epithelial stem cell transplant, the cornea is perfectly okay and vision has improved from 2/60 to 6/6. This is another 16-year-old male with again post-chemical injury. This is over a period of time. You can see within 1.5 years, the cornea is clear and the vision is perfectly fine. The second example was vitiligo. I think my previous speakers, they talk about a disease, vitiligo, which is a loss of, anybody says, what is the loss of cells in them? Melanocytes. It's a pigmentation. The routine care is you have to give a suction pressure on the thigh. And if there is a bulge, you cut it. These pieces are cut and then transplanted. The process is very painful to have epidermis, which you cut it and put it on the affected area. And sometimes you don't get enough skin from the donor site. So for that, one of my students who was doing PhD, he said, ma'am, our hair is black and it always grows. So there are certain pockets in the hair, which is called bulge stem cells. They give color and the new hair growth. So why can't we isolate those cells and see if they can turn into melanocytes? So Anil was the PhD student who we requested the dermatologist to give us few hair follicle and we grew them in the lab and they formed the whole sheet of bulb stem cells and we were able to differentiate them into melanocytes. So these are different star-like cells, which are truly melanocytes. So after that, we went into the transplantation with the help of Dr. Somesh Gupta, who is a dermatologist. And this is before and this is after. We undertook 15 cases as per the permission of the ethics committee, and we followed them for 4 years. And there was no loss of pigmentation in these patients. So there's a whole lot of clinical trial which we did using stem cells. This was all autologous because there's no ethical concern and there's no rejection because no need for matching. Now my lab is focusing on mesenchymal stem cells because most of the trial which is using stem cells, if you see clinicaltrial.gov, it's mesenchymal stem cells. So they have a certain properties. You need not go for the matching of these mesenchymal stem cells. You can pull together from 5 different donors and give it to one person, or I can give it to anybody without matching, or I can receive these mesenchymal stem cells from anybody without any matching. And they have a very high proliferative index also. So this is all listed here. I'll just go. So what is the mechanism of these mesenchymal stem cells? Number one, they secrete some proteins, cytokines and growth factor, which is called a paracrine effect. They themselves don't do anything. They secrete these factors, and these factors rescue some of the damaged or the dying cells. The second activity is they secrete small pockets of these small particles, which is called extracellular vesicles. They are very small, and they are packaged with high-grade microRNA, RNA, DNA, and also cytokines and growth factors. So these are the 2 modalities by which mesenchymal stem cells work. So as I said, that these extracellular vesicle or exosomes contain some cargo which is packaged by the parent cells, and they are as effective as the parent cells. So I just skip this one. So from the cells, we try to move towards the cell-free approach. That means can we leave the stem cells aside and use these extracellular vesicle as a drug? So this was our approach. So first question was, how do we isolate that? Because these extracellular vesicles are secreted in the conditioned media or the media. So this work, I gave it to one girl, Suchi, and she was having difficulty in isolating them. And she was very, very angry with me that why you have given me a PhD topic, not of stem cells, but also for the extracellular vesicle. But she's tried to understand that these extracellular vesicles are very small. We can use the virus isolation method to isolate these cells. So she used this method, and she was very successful in isolating these extracellular vesicle and standardize this technique, which is very well cited in the literature. Then we try to use these extracellular vesicle in different diseases. I will touch upon few different diseases. First is liver. So what is the damage which is caused to the liver is by different agent. It can be a virus, it can be unhealthy diets, junk food, or alcohol. In India, it is mostly the viral factor followed by the alcohol abuse, which can lead to chronic injury. And what happens in chronic injury, when the hepatocytes are injured, they secrete some of the pro-inflammatory cytokines and they activate the macrophages. After macrophages are activated, then they act on the hematopoietic stellate cells. These are the immune cells present in the liver. They activate and they create extra ECM, which is mainly collagen type 1 and 3. Some collagen is good, but extra collagen leads to the fibrosis. So this is the pathophysiology of the liver fibrosis. And this was what my student Suchi wanted to check. So what she did, she isolated extracellular vesicle from umbilical cord, derived cells, and also the adipose tissue derived mesenchymal stem cells. And she created an animal model of liver fibrosis. She introduced these two different type of extracellular vesicle. And then her final assessment was, how does hepatocyte proliferate? Is there a decrease in fibrotic marker? How does the ECM is behaving? And also the anti-inflammatory and the fibrosis score in the liver. So I will quickly go through some of the assessment assays. Before going into the animal, you need to prove your hypothesis in vitro. So in vitro, as I said, we need to check with hepatocytes and also the stellate cells. So she made an in vitro model of liver using these two different type of cells, namely hepatocytes and stellate cells. She first damaged them by giving different agent. Then she introduced the extracellular vesicle, and the endpoint was hepatocyte proliferation should be restored. There should be decrease in the reactive oxygen species, which is the ROS. And also in terms of stellate cells, ECM should be decreased, and also fibrosis scoring should be decreased. So yes, we got everything here in the in vitro system. We did the proliferation of hepatocytes were there, and we saw that there was reduction in the ROS, and also in the stellate cells, there was reduction in the fibrosis. So in vitro, fantastic. So what about in vivo? She first created the animal model, and she had this endpoint analysis, which I will go one by one. First, biochemical parameter is your liver function test. So she found that administration of EVs, they reduces the elevated liver function test, which is ALT and AST. And also the hallmark of our gold standard of any immune cells or any pathophysiology is your histopathology. The H&E staining, Masson trichome, and Cyrus, these are the all gold standard tests, which denoted, yes, there is a significant decrease in portal and lobular inflammation and fibrosis, which is indicative of that, yes, disease is getting reduced. And this is again the marker for different markers of liver fibrosis, which is listed here. And we found that there's a significant reduction in the expression of these marker, which is indicative of fibrosis. And there's an increase in regeneration marker, which is called PCNA positive nucleus present in the hepatocytes. Our next question was, okay, we are using EV and we are leaving behind the mesenchymal stem cells. So in my lab, there was a debate. One group was favoring stem cells and one group was favoring EVs. So we said, okay, let us see what is the distribution and homing of these stem cells versus these extracellular particles. So when we administered them in the animal model, we found that most of the MSCs are localized in the lungs. So this is the chart here. They are going into the liver, yes, but they are also going into the lungs and also into the spleen, whereas EVs are not going into the lungs that much. For example, if I give 100 MSCs, 40 are going into the lungs, 10 are going into spleens, and very less, 50 or 40 are going into the target organ, which is the liver, whereas the amount of EV which is going into the liver is more. And this is a 7-day data. We have done it at 14 and 21 days also. So the retention of EV in the target organ is much better as compared to the cells. So this was our finding. So next, we went into the brain, which is like if somebody's brain is damaged, what happens? So can EV cross blood-brain barrier? So this is the hindrance where the stem cells cannot cross the blood-brain barrier. So this was again our hypothesis. So we created a JEV, Japanese encephalitis virus model. This virus causes neurodegeneration in the brain. And this work was done with collaboration with Aru Banerjee from RCB. What we did, we isolated bone marrow, grew the MSCs, isolated EVs, and we first did again in vitro studies. C17.2 is a neuronal cell line. So what we did, we made neurosphere. So brain is not a single cell layer. So EV can penetrate single cell. So what about if there is a 3D structure? So EV has to penetrate different layers of the brain. So we made neurosphere and tried to introduce EV. And same way we wanted to see in the animal model. And we found that JV infected mice, they are like easily affected and there's a death. However, when we introduce EV in this infected animal, their death is less, the longer survival, and symptoms are also delayed. So in short, this is again in vitro analysis. I will not go into the dose determination because we need to know how much EV is needed. So this is a neurosphere model where the neurosphere is infected with JEV. You can see here control, the size of the neurosphere is big. If you infect this neurosphere with virus, the neurosphere is not developing. But when you introduce EV in them, that's a 10 microgram, 20 and 30, the neurosphere size is increasing. That means virus effect is being nullified by the EV. So this is again the same thing. So let's come to the in vitro. So in vitro, we created the JV virus model and we saw that their survival is increased. In the brain also, there is no presence of the neuronal protein, which is NS1. So here is EV, bone marrow. The virus load is less. And here also in the brain tissue, you can see the JV virus titer is less when EV is given. So this is a control animal on the left-hand side. In center is PBS. Third one is the infected. And the last one, which is very active, you can see it was infected and we gave the virus. So it is jumping out of the Petri dish. But I am not saying that it is all the animal were like that. This is the best animal which my student has tried to photograph. So basically, this is just brain slices also because we need to see other very thorough studies, what is happening in the brain. And we found that in the brain, there's a lot less virus and there's a better neurogenesis is there. So again, the same thing here. So what MSC EV is doing, it is anti-inflammatory. It can cross blood-brain barrier. It is neuroprotective. It is supporting neurogenesis there. And the symptom of this virus is being delayed. And there's a less death from this infected. There were few deaths, of course. So now we are increasing the dose of the EV because we gave the less dose. So another study is going on with a higher dose. So another third one and the second last one is about the skin. So the next question was, how do you enhance the properties of the EV? So what do I mean by enhancement is, for a particular dose, as I showed you in JV, from 10, we are increasing it to 30 microgram. So what if I increase the power of those 10 microgram of EV? So that I don't have to make more amount of EV. So 10 microgram having the power of 30, that was our plan. So can you tell me in what condition we culture these cells in the lab? What is the oxygen concentration? Anybody? I don't know. You are biology students? No? Okay. So in the incubator, the oxygen is the normal atmospheric oxygen. So what is normal atmospheric oxygen? None? 21. Yes, it is 21%. Okay. We culture all our cells in the incubator with 21% oxygen. So, but inside our body, where stem cells reside, the oxygen tension is less. Fine, it is between 2 to 8%. So that means we are not culturing our cells in their native state. So this was the hypothesis with which our students started working, giving this low oxygen to these cells. We gave around 2% to 5%. Suchi again worked on that, and she found that hypoxia preconditioning of the parent cells, it modulates EV. So these EVs are much better in terms of immunomodulation, regeneration, and their secretion from the cells is also increasing. So these are the things. I will not go into the detail. So then Yashvi took this forward. She find, okay, let me take these EVs, give them, compare normoxia versus hypoxia, what is happening. And then she did the multi-omic studies to found what is the cargo difference, RNA profile or proteomics, and can this be validated in the in vitro model and can it be made a product? So I'll just go quickly. She was working on skin regeneration model. In the skin, we have two types of cells, fibroblast and keratinocytes. So she did on them and she found that hypoxia preconditioning of the parent cells, not the EV, parent cells, give you the EV, which is much better in terms of migration of the cells and the proliferation of these two types of skin cells. Fine. So these are much better. So in terms of immunomodulation, she treated with lymphocytes from the patient and she found that these EV have better immunomodulatory properties. That means they are making your cells from pro-inflammatory to anti-inflammatory cells. So these are the two types of assays which is routinely done. I'll not go into the detail. Okay, so we have done this assay to enhance the cargo of the EV. So she said that let me see what is the main component difference between normoxic and hypoxic. So she published this paper and she did the proteomic analysis and she found that some of the regenerative and immunomodulatory proteins and microRNAs are very much enhanced in this EV. So she picked up two microRNA, which was very much high in those hypoxic one. MIR125 and MIR145. This is all published. And she did an assessment of this and she proved her hypothesis by silencing these one. I will not go into the detail. I think it is too exhaustive for you. And she validated this role of these two microRNA. And then she said, okay, why not I upregulated these two microRNA in the EV? Number one, to give hypoxic treatment to the parent cells and to increase these two microRNA inside the EV. So that was her hypothesis. So she did that and she then combined the two EVs, this one and this one, because MIR125 is having immunomodulation power and 145 is much more regenerative in terms of proliferation cells. Then again, she tested using two different cell lines in the lab and then went into the animal model. So this is her animal model, which is traumatic wound injury. It's called excision skin model. That means you take the chunk of the skin out and then they treat the animal. I will just skip this one. So you can see here when she gave the dual one, means where both the MIR is combined together, the recovery is starting from day 7 onwards. You can see here. Fine. And when you are downregulating these MIR, like you are suppressing, I means inhibited. This MIR is inhibited. That means the inflammation or the wound is not closing. That means that is their role is very much established. So she did further analysis. Everything, what she found that enhanced regenerative ability of dual MIR, that means combination of them, which results in better epithelialization, stromal regeneration, and also collagen fiber rearrangement. So if collagen fiber rearrangement is good, that means you can achieve scarless healing, which is not there in us adults, but it is present in the children, pediatric population. And she also found some few hair follicle. So that means this dual formation can be used for hair regeneration. So I think I will just skip this. This is a mechanism. I think I will not go into the detail. So she further, her thing was, she got a BIRAC big grant to see that how you can preserve these stem cells. As I said, to preserve cells, what do you need? Ultra low temperature. If your cells are dead, that means they are inactive. But these EVs, if they don't have nucleus, so that means their shelf life is much better. So using this BIRAC supported grant, she tried to study that, can I make a model? I think that slide is not here, at different temperature. So what she did, she kept these EVs at room temperature, 4 degree, -20, -80 for 1 month, 3 months, 6 months, and 9 months. And she studied all their biological and physical properties to check their stability. So everything of product development was done in the GMP lab. She's here, she's cryopreserving these cells here. And this is the product she has made in a powder form. And this is easily miscible using normal saline and also the PBS. Sorry, this is the one she did physical and functional exploration of these EVs at different temperature and time. Again, I will just leave this. This is all published. So what is the take-home message is hypoxia priming of the parent cells is a very minimal way to manipulate your cells. Wharton jelly EVs, they are superior to bone marrow, and combination of these EV can be considered for regulatory and regenerative properties. And she has patented this product. Another student, Suchi, which worked on liver, is also has made her startup, which is called TCIPL, and she has represented her company in multiple global BioIndia platforms. She will be now launching her project soon. Now we moved on to tissue engineering. This is my last. It's very easy to put this EV or any formulation on the skin. So what about where we need a longer retention of these EV at the site? Shruti thought about integrating EV with the tissue engineering. So in that, what she did, I will just skip this. She fabricated a scaffold which she named as SHE, which is a combination of silk, amniotic membrane, and EV. So this is the product, which is very sponge and very moldable. It can fold and it can take the counter of any wound or any body shape. Then she did again in vitro analysis as per the norm. And she did the in vivo analysis in the skin wound model. Again, you can see here, duoderm, which is commercially available collagen flap, is taken as a control. And she is again showing very good regeneration from day 7 onwards. So she has done the mechanism, which is IL-6 mediated, and also reduction in the inflammation. And the wound closure is much better using this. Again, this one is patented. And now she's also going for a third-party validation in the large animal, because she has done in the rat. She's going to do that in the porcine model. So what is the future work which we are trying to do, which can help in the product development, is use of 3D printing. One student, Shruti, is working on 3D printing. Shruti and Chippy working on 3D printed skin. And also Dr. Somya, she is working on 3D printed bone modules. This is again, this is the whole bone, which marrow and the cortical bone, which is using 3D printing machine. So this is where we are. And for every research which I have presented, there has to be certain guidelines. Regarding that, for stem cells, we have ICMR DBT national guidelines to follow for stem cell based research. And for extracellular vesicle, right now in India, we do not have any guidelines, but there is International Society for Stem Cell Research and also international guidelines, for which I am part of that, and we are formulating it. And also this is International Society for Cell and Gene Therapy. There also we are formulating some guidelines regarding exosomes and extracellular vesicles. So the point is, this field, EV field, is very promising, but still it is evolving. We need to know the biology of this EV more. I have only pointed out microRNA. As I said, there are a lot of proteins. We do not know what hypoxia is doing to those proteins. We need to know more about this biology. EV isolation, purity, potency, and heterogeneity, we need to take care. And also large-scale production is again a bottleneck. So last but not the least, I would like to thank all my students. Suchi working on liver model. Sonali is the girl who worked on the hypoxia thing. Yashvi on the skin and the dual microRNA study on the skin. Shruti on the SHE scaffold. Somya is working on integrating bone explant and bone scaffold using extracellular vesicles. And also AIMS faculty from pathology, ophthalmology, dermatology, and all the facilities of the AIMS. Arup and Anupam from ILBS and RCB for the liver work, and also for the JEV work, all the funding agency, and of course the whole team which has contributed in some way or the other. I would like to say that we have our Indian Society for Extracellular Vesicle, which had last year inaugural Evolve annual meeting. This year Evolve meeting will be in Bhubaneswar Institute of Life Sciences. You all are welcome to join the society and join this conference. Thank you all.
Thank you very much, Dr. Sujata ji. You have really taken us to the next gen therapeutics segment. After a long time, we have been listening on stem cell therapy, though we are only knowing it the superficially or just the outline, but how it is applicable on different physiological in vitro, in vivo aspects. We have really learned a lot from your talk. And I'm very sure that all our faculty and students, to some extent, they have definitely understood it. It's not merely a lecture. I think stem cell, it's a big encyclopedia. We are to enter into that, and as you mentioned, still it is evolving. So we are very sure that in the time to come, we'll definitely have the better applications.
Thank you,
Once
sir.
again, thank you very much. Thanks.
Thank you, ma'am, for your highly insightful and informative presentation. I kindly request ma'am to come on stage and for the session chairs to facilitate ma'am.
Recharge the students for that. I think everyone in the audience is recharged.
I would like to call upon stage Dr. Ankush Pandey. I would like to call upon stage Dr. Ankush Pandey, Director, Regular Affairs and Quality of PRG Pharma. I'll request the session chairs to facilitate Dr. Ankush, sir. Thank you so much, Dr. Ankush, sir. I'll request the session chairs to kindly introduce the fourth speaker.
Good afternoon, everyone. It is my pleasure to introduce our speaker, Dr. Nitin Sharma, Head of the Department, Pharmaceutics, Amity Institute of Pharmacy. He's a distinguished pharmaceutical scientist with over 18 years of experience in drug delivery systems, nuclear imaging, and translational research. His contributions to bridging academic research and industrial applications are highly commendable. Please join me in welcoming Dr. Nitin Sharma to deliver his presentation. Dr. Sharma, the floor is yours.
ऑडियो या वीडियो है
Good evening, everyone. So thank you once again on the chair, Dr. Khadagwal, Dr. Sharma, and Dr. Pathak. And thank you very much once again for the wonderful opportunity. So my presentation would be depend on the largely, we all are working on the pharmacy. Some of us, we are synthesizing the drug in the laboratory. Some of us are finding their therapeutic value. Some are working on the toxicity aspect. I am working on the formulation aspect, means how we can improve the bioavailability, how we can target the drug to that delivery site. So this is the topic. I would like to appreciate the organization committee because we are running ahead of the time. So I will summarize my presentation, very concise, but this 15 or the 30-minute presentation would be full of the interesting fact how we can find out whether our concept is really working in the in vivo model. So whatever we are developing in the laboratory, regardless of the kind of the formulation, we are working on the tablet, the capsule, the ointment, the syrups, the nanoparticle, the resealed erythrocytes, and my previous speakers are working on the EVs. That are again the mode of the targeting aspect. So whatever we are developing, whether it is going to the targeted site, that finding is really, really very important. So is it possible to look after the administration, how the formulation is traveling inside the body? Really, this is the traveling. This is the journey of a tablet. This is the journey of the formulation. So is it possible? Yes, this is possible. And the current technology, which is called the scintigraphy technique, the radiopharmaceutical technique, made this possible in the current era. And the technique is similar as our parents are tracking to us through our mobile phone or through the GPS. Whenever we are traveling, whether we are in the campus or outside the campus or in the market or enjoying the party any malls. So every time, if you're given the access to your parents can monitor you every time. In the same way, we can monitor the drug or the formulation. But there, the mobile is not, the formulation is not carrying the mobile. The formulation is carrying the radioactive material with them. So whenever the formulation is going, along with that, the radiopharmaceutical substance are going, and from the external camera, we are capturing the images. So this is a wonderful technology to find out whether our concept is working or not. Right? So, as I told you, we are working on the biodistribution. So this is the general aspect, what is the biodistribution? This is how the drug product or the formulation is distributing into the different part of the body. Why it is so necessary? Because every part are having their distinguished characteristics. The bone tissues are very, very strong, and that the distribution process would be very difficult. Unlike to the tissue of the liver, because the liver tissue are very soft and the distribution would be very easy. So at which rate the drug or the formulation are distributing into the body. So this technique is called the biodistribution, and the factor influencing the biodistribution is the physicochemical property, the biological factor. These are the blood flow, the tissue permeability, the binding of the plasma protein, and especially the route of administration, at which route the drug or the formulation is going to be administered. So these are the basic traditional methodology of the biodistribution determination. So what we are doing here, not we, the traditionally the methodology is we are delivering the formulation of the drug to the any animal model, and in the traditional methodology, that animal need to be sacrificed, and after sacrification, we are isolating the organs, and these organs will give the single suspension method, and there we are finding out the quantity of the drugs, how much quantity are distributing the different organ, right? So through this traditional methodology, we are finding, but these traditional methodology are having some short of the limitation, like it is an invasive method, the labor intensive, the time consuming methodology, sample variation maybe they are like I am not the skill of the withdrawing the blood sample. So my blood sample withdrawal methods would be entirely different to the specialized person. And lastly, the quantification technology, that is again the hindrance of the barrier for the finding out how much quantity of the drug is present in the particular tissue. So these are some limitation of the traditional methodology. What is the radiopharmaceutics? We are already gone in the entire B Pharma syllabus. The radiopharmaceutical substances they are regularly emitting some sort of the radiation, the gamma radiation, the alpha and the beta radiation. So these radiation we are using in this technology. So this is the camera which can be used for the determination of this fate of the formulation, where the formulation is traveling. Now, let me tell you what is the concept. This concept is similar to the concept of tracking. The way our parents are tracking to us, they are keeping the GPS in our bag. They are keeping the mobile phone in our pockets. Similarly, we are taking the formulation with the radioactive material. So we are making the formulation of the drug labeled with a radioactive material. And this will call the radioactive drug. Now, whatever your formulation is, here I am taking the example of the nanoparticle. So whatever the formulation, it could be the tablet, ointment, syrup, capsule, whatever the formulation. So these radioactive formulation or the drug would be entrapped in that particular formulation. Now this will become the radio labeled drug embedded in the drug delivery system. So once this complex will be developed, so this will be administered into the any animal model or any human being, and through this gamma camera, which I already shown in a previous slide. So through which this gamma camera, we can taken up these kind of the image, which is called the gamma scintigraphy image. And these kind of the images are showing the clear picture where my administered drug is going inside the body. See, in this image, this is the site of administration at the left arm of the human being. And after the administration through the IV route, the larger your drug is going deposit into the liver. So this is the liver. The some amount is here in the heart because the drug was administered through the IV route. So definitely the drug, the largely amount is going to the heart. And there are some traces of the amount of the drug into the kidney because there is some parallel excretion of the drug. So the drug is available largely into the kidney. So these are the two kidney, right kidney and the left kidney here. And after the excretion, the drug is, where the drug is now? Can anyone tell this, what is the organ this is? This is the bladder, urinary bladder. Now the drug is ready for the excretion. All right. So if we are capturing these kind of the image at different time point means immediately after the administration, at 1 hour, 2 hour, 3 hours, so we can find out where your drug is actually right now in the body. So this is the technique which is called the determination of the biodistribution. Now, again, the next slide is showing the different kind of the radionuclei. So these are the formulation. We can label the drug, we can label the formulation, and within the formulation, we can label the microsphere, the nanoparticle, the liposomes, the DNA, and the EVs, which are currently being used for the targeting purpose. And after the labeling, we are administering in the animal model or in the human being. And this is the gamma camera. And after capturing the image, so these kind of the image we are capturing in the gamma scintigraphy techniques. I'm not going into the detail of these different kind of the radioactive materials. I'm skipping. So I'm directly coming to the case study, which we have done in the laboratory, not here in the Amity, but these, whatever the slide I'm showing here, these are the part which we have done in the INMAS, which is this is the DRDO laboratory. And the few part is performed in the laboratory of some private, the pharmaceutical industries, means this was the consultancy project. So in oral formulation, we can find out the disintegration time, the gastric emptying time, or the transit time of the formulation, like after the administration through the oral route, where the exactly tablet is in the stomach, how long it is going in the retain into the stomach, and at which rate it is traveling into the duodenum, jejunum, ileum, ileocecal junction, and how long it is going to be distribute the drug into the colon. So this is the entire journey. Similarly, we can find out the drug release behavior, the mucodation. We are already working on the mucodesive microsphere, mucodesive polymer, mucodesive tablet in case of the GRDADS, gastroretentive drug delivery system. So whatever the formulation or the concept we are working, whether it is actually following the same mechanism which we expected in the human body after their administration, then we can find out. The effect of the food, effect of the type of the food, the high fat diet or the low fat diet, the pre-food condition, after food condition, what is the traveling influence or what is the influence of this food on the transit time or the mortality on the formulation that we can find out. All right, so here we developed the sustained delivery formulation, and this sustained delivery was coated with the cellulose acetate phthalate, which is a commonly used polymer for the entry-coated tablet. So this is the requirement that whatever the formulation we are developing, that should be remain stable into the stomach, or there should be the least amount of the drug should be come out from the tablet when it is remaining in the stomach. But immediately when it is traveling into the duodenum, jejunum, ileum, or the small intestine, the drug should be released. So this was the concept. So we took the drug, we coated with the cellulose acetate phthalate. This is a pH specific polymer. And after the administration, and this whole work was after the due permission of the ethical committee. So these were the capture images. So this is the image at the 5-minute and the consecutive images. So the tablet is still in the colon, but very least amount of the drug was coming out. because here the drug was labeled. So you can have the evidence, whatever the amount of the drug is releasing out of the tablet. So this large or the intense image is your drug. Oh, sorry, this is the tablet. And around the tablet, this is the release amount of the drug. And through this technique, this is also possible ki how much about the drug is coming out of the tablet. And how it is possible? Through the calculation, which is called the region of interest. So here we can find out ki whatever the amount of the radiation is coming out of the tablet. So this is the 100% we are considering. This is the 100% of the drug. And amount of the radiation which are coming at this area. So this will be nullified from the amount of the radiation from this tablet. So through this methodology, we can find out how much percentage of the drug is coming out of the tablet. So here we developed the colon specific tablets. This was the work of my PhD student. So here we made, this was the mesylagine tablet. We coat with the polysaccharide, and the concept was because our colon is filled with the various kind of the microbiota. So whenever the tablet will go inside the colon, so the microbiota will consume the polysaccharide and all the drug will come out. So in this process, whether my concept is working, so we radio label the mesylagine and coat this mesylagine tablet with the polysaccharide and capture this image after the administration. So this is the image of at the 1 hour. So it is showing that the tablet is still in the stomach. At 15 minutes, the tablet travel to out of the stomach. At 30 minutes, it is in the small intestine. And after 600 minutes, or even before, at 420 minutes, the tablet is at the ileocecal junction. Right? So this is the ileocecal junction. Here the tablet, at this time point, the tablet was intact, largely intact, and there was the very least amount of the drug was coming out of the tablet. But immediately when the tablet is coming at the ileocecal junction, so the concept, as per the concept, the tablet was performing, means my polysaccharide was largely consumed by the microbiota present on the colon, in the colon, and all the drug was coming out. And this is showing through this image because all the colon was filled with the radioactive drug. So this was the validation, real-time validation of my concept. Oh, this is the proof of the concept by the imaging technology. Similarly, in the pulmonary and the laser product, we can find out the lung deposition pattern, how the developed formulation like the MDP or the tripod or inhaler we are preparing. So at which rate it is going to be deposited in the lungs, or actually at which site of the lungs or which area of the lungs the formulation is going to be deposited. Similarly, the drug residential time, why it is important? Because most of the time the nasal formulation or the lungs experience the mucociliary clearance. Whenever the formulation are going to be administered into the lungs or the nasal cavity, immediately the mucociliary clearance will start working and a largely amount will be eliminated from the nasal cavity or the lungs. So on that area, we has to be used some kind of the concept. This could be the enhancement of the viscosity of the formulation, or we can use the mucoadhesive polymer. So whether our mucoadhesive approach are working or not, so that we can find out with the help of this technology. Similarly, the product performance and the device and the spacer performance. We are having the multiple kind of the device for the single product. Let me show you the examples. So these were the gamma scintigraphy image. Here we radio label the formulation, not the drug. Here the formulation was labeled and these kind of the image was captured. So these images are showing, the lateral image are showing the here. This is the percentage of the amount available at the stomach, and this is the percentage of the amount available at the lung side. Similarly, this was the image. Here we convert the salbutamol. This is the bronchodilator. This salbutamol was converted into the two different formulations. One was the microsalbutamol and another was the nanosalbutamol. And after the administration in a single human being, we capture this image. So here in the microsalbutamol, the 25% was available at the upper pulmonary route and very small quantity was available at, means the 14% was available at the lung side. But in case of the nanosalbutamol, the larger amount, 81% of the drug was available into the lungs and very least quantity was available at the upper part of the pulmonary. So this is the clear-cut indication. What indication? That my nano formulation are really working, or that the pattern or the aerodynamic behavior of the nanoparticle are really actually working. Similarly, this single compound, the nanosalbutamol, was delivered into by the three different kind of the device. So these are the image. Here, this product was delivered through the device 1, again the device 2 and the device 3. Can anyone tell me which device are showing the best performance here? Device 3, exactly. So because here we can have the image largely your whatever the drug or whatever the formulation you developed, it is equally or the uniformly distributing into the lungs, and this was the objective. Similarly, the effect of our approach means whatever the approach we have used, the mucoadhesive polymer or the enhancement of the viscosity, whether it is still working against the mucociliary clearance or not. As I told you, whenever you will deliver the drug or the formulation into the nasal cavity or the lungs, so immediately after the administration, it will drain out. This is the response of the mucociliary clearance. So here we administer the formulation into the human being, and at the 360 minute, the consecutive images was captured, and we can have the evidence of the formulation are still available into the human being. Now, the effect of the nasal formulation, because again the nasal cavity, the vestibules, turbinate, and the atrium region, this is the anatomy of your nasal cavity. At the turbinate region, again there is the mucociliary clearance. So there also these special kind of the approach are required. So whatever the approach, regardless the type of the approach, whether it is working inside the nasal cavity after their administration, we can find out these kind of the result in the gamma scintigraphy technique. Similarly, we perform the topical product, and here in case of the topical product, we can find out the topical permeation and the topical penetration, the skin deposition and the localization, the effect of the transdermal drug delivery patches, the PK data of the different kind of the product, regardless to kind of the form, the indication, it can be used for the wound or the burn. So these are again the images which are showing the gradual decreasing or the absorption, decreasement of the radioactive material means where it is going inside the skin. So the rate of the penetration of the permeation we can find out with the help of this. So similar kind of the image we captured when we applied the topical product over the hand. So these were the some result where we target some kind of the targeting efficiency of the product. But apart from this, we can find out the anti-inflammatory potential and the antibiotic action. So I'm directly going to the antibiotic action because this result was very, this was a tedious job. So what we perform, there is a mechanism of the WBC, they will accumulate at the infection site. So instead of labeling the drug or the formulation, here we label the WBC. All right. So in the before pre-treatment and the after treatment, so before the treatment, this autologous WBC was isolated, and after the labeling, it was again administered and the image was captured. So this is the image at the 2R of the pre-treatment, and this was the image of after the treatment of the patient by the antibiotic. So in this image, we can have the larger amount of the lesion formation, and this lesion formation in the UTI. So what is the indication that after the treatment, this UTI is going to be suppressed? So this is a clear-cut indication that your antibiotic therapy is really working. And this is possible with this help of this, the visualization technique. Similarly, in the form of the oral product, we can find out the ocular retention or the corneal penetration, the ocular pharmacokinetic, the quantitative drug analysis, and the nasolacrimal excretion. Means this is the similar concept. What are the amount we are administering here in the cul-de-sac? This is the cul-de-sac, right? So there is a technique of the administration of the drug into the ocular region. Once you will administer, so largely amount will be drained out through the nasolacrimal process. So for that, or to minimize this drainage, we need to use some methodology. So this methodology could be anything, the mucoadhesive or the nanoparticle approach, or the electrophoresis. So whether your technique is working or not, so that we can find out with the help of this ocular product. However, our laboratories are not working on this laboratory button because the concept is same. So we can also work on the stage, the open, and we can also explore how this technology can be useful in this ocular product as well. So these were the mucoadhesive action we perform in the laboratory. So the different time point that image was taken, and these are clearly indicating the formulation was available into the cornea or into this ocular cavity. So these were some recent publication which we did with the PhD students or my lab mate in the INMAS or with the private company. So this first, the illuminating the inside the clinical study harnessing the pharmacoscintigraphy. This was the consultancy product which was taken by my laboratory of the very good name or the very big brand. This is the diclofenac gel, and here we perform the publication was on the naloxone solid lipid nanoparticle. Similarly, these are the few of the publication which we made with the help of this gamma scintigraphy technique. So I would like to thanks my mentor, Dr. Singh Bhatnagar. He is extreme right and extreme left is myself. So I really thankful to my mentor, Dr. Bhatnagar. He blessed me with this technology, and in between Dr. Bhatnagar and me, there's Koshagar and the Neeraj. Both was my lab mate, and we are now working on this gamma scintigraphy and helping the pharmaceutical industries to find out the real-time evidence ki how their product is working actually inside the body after the administration. So thank you very much. However, I try a lot to make my presentation easy and the concise to limit it to the time. But still, if you are having any doubt or the question, I would like to help. Thank you.
Thank you, Dr. Nitin Sharma. You have taken up a very important aspect, that is the drug distribution in the different dosage forms, elaborated the mechanism, how it would be readily available for the body through the oral route, topical route, parenteral route, even the ocular and nasal preparations. I hope that we all have taken up the important contents from your presentation. Thank you once again. If anybody have any query from the audience relating to this presentation or maybe for earlier also, you all are welcome. Please. Thank you, Dr. Nitin.
Thank you.
Please.
I hope I'm a good teacher.
Yes.
Thank you, sir, for your valuable contribution. Yes, sir.
Some student want to ask some.
Question?
Either you come forward or you provide the mic to them.
गुड इवनिंग सर। सर मेरा क्वेश्चन ये है कि जो एंटी रेबीज इंजेक्शन होते हैं वो इंट्राडर्मल दिए जाते हैं, इसके पीछे क्या रीजन है सर?
This question is out of my scope of the topic. However, I would like to attempt, because this is the intradermal. So the route of delivery system are largely depend on how your drug molecule are traveling or how your drug molecule is absorbing into the body. So sometime it is necessary to deliver through the IV route, because IV route is going directly into the bloodstream and is showing the 100% bioavailability. But in some case, if you want your drug or your therapeutic substance should not be directly administered into the body, because there should be some incubation period before their administration. So we need to hold the drug. I hope, or I guess, this could be the reason of delivering the anti-rabies drug through the intradermal. However, this is the question taken up by the pharmacology. This is not my area. So your pharmacology teacher will justify this answer, whether I'm right or wrong.
Thank you, sir.
Thank you.
Yes, thank you.
Come forward.
In case of nano formulation, you told that there was a too much deposition of drugs were there. So don't you think too much deposition of drugs in the lungs, what is shown in the slides, will cause toxicity specifically in lungs and stuff? So we also need to perform the toxicological aspect as well beside that one. And even it can cause like the radioactivity effect on that specific drug. So how you are going to assess that one?
Okay, whatever that I'm getting, so you have the two questions. One is the toxicity aspect of the nanoformulation and another the toxicity aspect of the radio substance. And before taking up your question, I discussed with her that this question will become. Definitely. So I am attempting your first question, that nanoformulation toxicity. Definitely the toxicity is associated with everything. As the Ayurveda speaker told you, none of the formulation, this Ayurvedic formulation, we cannot consider as a safe. The Haba formulation are also the toxic. In the same way, the nano formulation are also the toxic, because these are having the very minute size or tiny size. They can travel everywhere into the body, where even your toxic substance cannot be. Okay. So for that, so this is the good side of the nanoparticle. So we are taking this good side of the nanoparticle and we are decorating the nanoparticle in such a way. This is called the nano engineering, or we are modifying their toxicity aspect in such a way that we are dealing. We will have only the therapeutic value. Now we are not going to have the toxic aspect. One thing. Second, the toxicity are related to their size specificity. Size. If your nanoparticle will have that certain shape or the shape, the size, it will go to that particular location or the particular area and will show the toxicity only. But if your particle are beyond that or above that size, so it will not show any toxicity. But largely we are modulating the nanoparticle with the polyethylene glycol and several another type of the material. It will not be the toxic at that level, which it is showing the toxicity, the prior not to be the still. It is called the stealth nanoparticle. And your second question is related to the toxicity aspect of the radioactive material. So, my dear, the radioactive material. So I told you, so this technology is not for everyone. We are not going exposed to the all human being with this aspect. So in case of when you are exposing to the X-ray, there we are exposing about the 500 millicurie of the radiation. But in this technology, we are reducing this radiation lower to 25 millicurie. So now you can compare. There is the 25 millicurie, and in case of that X-ray, this is the 500 millicurie. So you can consider. I am again in the favor of this gamma scintigraphy because this is my forte. But whatever the technology, they are having also the negative aspect as well. So we are working on that as well. So I'm not saying that this technology is completely safe. Yes, there may be some chances, but these chances are lower than the chances of the possibility of the radiation by your mobile phone which you are carrying. Even this is also the exposure of the radiation. We know that all thing, but then only we are carrying this because it is having some advantage. All right. So in the same way, this technology are also having some advantage. So we are taking that point and we are working on the negative aspect of this. And this all technology or the radiopharmaceuticals are the FDA approved and regularly being used in the diagnostic center. The technician, the indium, the iodine. When the doctor will prescribe you the CT scan or the PET imaging, definitely you need to be exposed with this technetium, whatever the substance is that we are already using the gamma scintigraphy.
Right. Thank you.
Thank you, sir.
For resolving issues. So thanks for your presence.
Thank you, sir, for your valuable insightful. I kindly request these session chairs to facilitate Dr. Nitin, sir. So many times. There's one, and then that's it. We have Dr. Surbhi Johri, Head of Department, Associate Professor of IMS Ghaziabad. So I would like to call upon you on stage. I request the session chairs to please facilitate our special guests for today. I kindly request Dr. Chitme sir to facilitate one of our session chairs, Dr. Anil Sharma.
Thank you so much, sir. Please come back. Yes. Come back.
Thank you. Yeah. It was so nice. It was nice to hear all the.
Connected so well.
Yeah. It was a pleasure.
I kindly request Dr. Rajiv Kharab sir. I kindly request Dr. Rajiv Kharab sir to felicitate Dr. Malika Pathak ma'am. I would like to call upon stage Dr. Nirupma Diman ma'am to facilitate Dr. Harsha Khalkarwal ma'am. With this, we conclude our day one of the conference. We sincerely thank all our distinguished speakers, session chairs, and participants for their valuable contributions today. We look forward to welcoming you guys on the day two of the conference. Thank you and have a pleasant evening. All the delegates.